recording. Okay. And this is getting out that this is not the way you play, but the way the squares are going to be, did you see the color squares? Like, did you see, so triangle is A, that kind of, yeah, that's what you saw, but you didn't see answers. Ah, that's getting on my nerves. Okay. All right, so you saw red, you see a red square, a blue square, whatever. And then the circle is C, and then the square is always D. So the triangle is always going to be A, the diamond is going to be B, the circle is going to be C, and the square is going to be D. And I'll always read out the answers. But this is no fun playing this way. But I am recording it. Because I don't know why you can't see it. So that's the problem. Okay. So, that being said, are you guys, okay, and I'm going to tell you the answer. So, let me just tell you what the squares and the circles represent. And then I'm going to record this so you can go back and look. But then you can go back in to play this game yourself, and hopefully you can see it. Okay, the red triangle is always going to be A, or the triangle will be A. Okay. The diamond will be B, and my diamond is blue. I think, oh, let me see. I think you have to show the question on a big screen. Oh! That's why this isn't working, and I can't show it. That's why you guys can't see each other's names. And you're right, because when I play it, I show it on a big screen. Okay, so this is the way we're going to do it. I'm going to read the answers, and you just pick the color and the square, and I'll tell you what's correct, okay? Is that okay for right now? So we can kind of take a quiz and just kind of, because I don't know how else to do it. So let's just try it and see how it works. All right, and then I'm always going to read it out. So let's go. So, all right. So the triangle, a red triangle is always going to be A. The blue diamond is going to be B. The gold circle is always going to be C. And the green square is always going to be D. All right. Does everybody have that? What I'm going to do is go right to the next question, but I'll tell you the answer. Now, I hope it won't reset itself. Let's see how it works when I go to the second question. So I'm going to read the first question again, and the answer to the first question is neurology, which would have been A. Okay. All right, let's try this. Oh, man. Okay. So the question was the diagnosis and treatment of nervous system disorders. And the answer was A, neurology, which is the red triangle. All right? So I had 3 people to get that right. And A-Bib, you're in the lead right now, okay? Because you must have answered quicker than anybody else. Okay, Lillian, you came in second. All right, let's go to the next. Okay. The next question. Loss of sensory motor, speech, language functions due to interrupted blood supply to the brain. A, multiple sclerosis. B, traumatic brain injury. C, gold circle, cerebral vascular accident. Green square, Pick's disease. Okay, so I had 8 people to pick C, cerebral vascular accident, and you are correct. Loss of sensory motor, speech language functions due to interrupted blood supply to the brain. The answer is C, cerebral vascular accident. And they're changing up the colors. So, but the shapes are the same. A is going to always be the, what do you call it? The triangle. B will be the diamond, C will be the circle, and D will be the square. Okay, so 8 people got that right that I can see, but not everybody answered. Okay, all right. Now, if you want to talk to me, just talk using your mic because I cannot see the classroom. Okay, she hadn't even read the answers before mine ran out. So, okay, so let me see. So that means this game isn't working really great. That, but it marked me wrong. Okay. Man, this is getting on my nerves. All right, so what I'm going to do is this. Okay, so what I'm going to do with this so we can get through the questions, I'm going to read the question and type say the answers. And then you can answer there, but if you want to, you can always answer it's going to be hard to answer in the box down, but you can always type the answer down if you want to. But I'm going to tell you the right answer, okay? I'm so sorry my Cahoots isn't working, but this is the first time I'm trying it. Okay. Yeah, we're going to try it, but let's get through the questions because it will definitely help you on your test. Okay, so just wait. Are you guys, let's just try. You ready? All right. Listen carefully. Here's the next one. Okay. Angela, right now, you're in the lead for some reason. Here's the question. These cells form myelin sheaths around axons in the peripheral nervous system. Ependymol cells, A. Astrocytes, B. Schwann cells, C. Oligodendrocytes, D. Ependymol, A. Astrocytes, B. Schwann, and oligodendrocytes. Okay. So these cells form myelin sheaths around axons in the PNS. The answer is Schwann cells, and that is correct. So we have 15 people to say C. You are correct. Okay, even though I think I checked the wrong answer. Okay, next. So this time, Lake and York, and you're first, and Michelle is second, and A. Bibb is third, and Angela is fourth, and Rachel is fifth. That must be the way you're answering in terms of the quickness. Okay, you guys ready for the next one? Okay, if it marked you wrong, just take this as a learning thing. So if you got it right, yay. Okay. But the reason it marked you wrong is because I think I marked the wrong answer in there. But if you got Schwann cell correct, then that is the right answer. All right, let's go to the next one. Okay, listen up. Here it comes. Okay, these cells have many dendrites and one axon. A, unipolar. B, bipolar. C, interneuron. D, multipolar cells. These cells have many dendrites and one axon. Unipolar, bipolar, interneuron. 23 correct. 23 answered. And the answer is multipolar cells. Multipolar cells have many dendrites and one axon. Okay. All right. And anybody have a question? Remember, raise your hand and talk because I cannot see Blackboard Collaborate. I'm right in the game. So, and I'm recording this so you can go back and listen to the questions and listen to the answers. Okay, we're going to the next one. All right. So right now, for some reason, Lakin, you're in the lead. Okay, next. These cells contribute to the blood-brain barrier. A, microglia cells. B, astrocytes. C, ependymol cells. D, oligodendrocyte cells. They contribute to the blood-brain barrier. A, microglia, B, astrocytes, C, ependymal, D, oligodendrocytes. And I've got the answer is astrocyte cells. So if you said astrocyte cells, you are correct. It's showing 9 people said that, but more people probably said that. These cells contribute to the blood-brain barrier, and it is B, astrocyte cells. Okay, next. Lakin, you're still in the lead, but we're not going to worry about the lead. Okay, here's the question, guys. The space between the presynaptic terminal and the postsynaptic terminal is A, synapse, B, dendrite, C, cell body, D, axon. The space between the presynaptic terminal and the postsynaptic cell. A, syntax, B, dendrite, C, cell body, D, axon. And the answer is A, synapse. And 20 people got that one correct. Okay. So right now, Kendra, you're in the lead. Lillian, you're second. Natalie, you're third. Amanda, fourth. And Lakin, you're fifth, according to the screen. Okay, and 4 players just hit the answer for some reason, but that's okay. If you got it right, more power to you. Now keep a running total of the ones that you have gotten correct so you can tell me after we get through this. Okay, we're going to the next one. The next question. Sensory and motor nerves that innervate visceral organs and glands. A, somatic nervous system. B, central nervous system. C, cranial nervous system. D, autonomic nervous system. Sensory and motor nerves that innervate visceral organs and glands. Okay, so the answer is autonomic nervous system. The autonomic nervous system innervates visceral organs and glands, so it makes you either fight or run away, okay, when you're afraid. All right, so right now, let's see. Kendra, you're still in the lead with Lillian, Julie, Natalie, and Michelle coming next according to this screen. Okay, again, keep a running total of the ones you get correct. Next, serves higher mental functions, integrates all sensory motor functions and perceptual experiences. A, the spinal cord. B, the brainstem. D, the cerebrum. C, the cerebrum. D, the cerebellum. Serves higher mental functions, integrates all sensory motor functions and peripheral experiences. The answer is C, cerebrum. The cerebral cortex serves higher mental functions, integrates all sensory motor functions, as well as perceptual experiences. Okay, Julie, you're in the first place. Kendra second, Natalie third, Amanda fourth, and Lillian fifth. Next. Mass of neurons, usually deep in the brain. Mass of neurons, usually deep in the brain. A, nucleus. B, tract. C, ganglion. D, nerve. A, nucleus. B, tract. C, ganglion. D, nerve. Mass of neurons usually deep in the brain. And the answer is A, nucleus. All right. Ganglion is a peripheral nervous system term. Okay. And all of this is coming from chapter 1. So we'll go over the ones that you have questions about. Okay. All right. So let's move on to the next one. But the answer to that, mass of neurons usually deep in the brain, is A, nucleus. Okay. Julie, you're still in first with Kendra, Natalie, Michelle, and Amanda following. All right. Next. serves as the power supply to the nerve cells by generating ATP to keep the cell alive. A, ribosomes. B, lysosomes. C, Golgi apparatus. D, mitochondria. Serves as the power supply to the nerve cell by generating ATP to keep the cell alive. The answer is, and most people got this, D, mitochondria. Mitochondria serves as the power supply to the nerve cell by generating ATP to keep the cell alive. Okay. Julie, you still in front. Kendra, Michelle, you moved up. Natalie and Amanda. Of course, this is probably not accurate because I know you guys are answering and it might not be recording. All right, the next one. Okay. These cells travel to the site of lesion and engulf cellular debris before removing it. Astrocytes, A. Ependymal cells, B. Microglia cells, C. Schwann cells, D. These cells travel to the site of lesion and engulf cellular debris before removing it. And the answer is C, microglia cells. Microglia cells travel to the site of lesion and engulf cellular debris before removing it. They clean up. Okay. All right. So I have 14 people according to this to answer it correctly. Kendra, you moved to the front. Natalie second, Julie third, Kiana fourth, and Amanda fifth. Okay. All right, we're on question 12 of 20. Glia cells of the peripheral nervous system are A, oligodendroid cells, Schwann cells, B, astrocytes, C, microglia cells, D. These are glia cells of the peripheral nervous system. Oligodendrocyte, A, Schwann cells, B, astrocytes, C, and microglia cells, D. And the answer is Schwann cells, B. Okay? Kendra's still in the lead. Kiana, Julie, Natalie, and then Lakin. Okay. Next. Vertical section into front and back. A, coronal. B, sagittal. C, lateral. D, transverse. Okay? Vertical section into front and back. A, coronal. B, sagittal. C, lateral. D, transverse. And everybody got that almost. The answer is coronal. Kendra's still in the front. Kiana, Julie, Natalie, and Amanda. Now I don't know how accurate these things are, so keep a running total of your responses. Okay. Next, 14 of 20. Back of brain or head. A, dorsal. B, rostral. C, ventral. D, caudal. Back of brain or head. A, dorsal. B, rostral. C, ventral. D, caudal. Okay, and the answer is caudal. Caudal is the back of the brain or head. Dorsal is the top. A lot of people said dorsal, but dorsal is the top of the head or superior. Okay, caudal is the back or towards the tail. Okay, so think of the brain. All right. Okay. All right, let's move. So let's see. Kendra's still in first place. Kiana, Julie, Natalie, and then Alyssa. Okay, next. All right, 15 of 20. Homologous are the same areas of the two hemispheres are connected through what types of fibers? A, association fibers. B, projection fibers. C, ventricular fibers. D, commissural fibers. A, association. B, projection. D, ventricular. And C, commissural. And the answer is, not E, no, C, D, commissural. So let me read it again. Homologous are same, it means the same. Areas of the two hemispheres are connected through what types of fibers? And the answer is D, commissural fibers. Okay. Alrighty, let's move on. Kendra, still in front. Well, yes, and the other things, other people still following in the same order. 16. The brain's ability to change as a result of experience. A, the centrality of the central nervous system. B, plasticity in the brain. C, laterality of brain organization. D, topographical representation. The brain's ability to change as a result of experience. The answer is B, plasticity in the brain. Excellent. Most people got that. Now, Kendra, you still in front. Kiana second, Alyssa third, Natalie, and then Julie. Okay, next. We are 17 of 20. Deals with the nature, cause, and diagnosis of diseased tissue in the brain and spinal cord. A, neuropathology. B, neurology. C, neuroanatomy. D, neurophysiology. Deals with the nature, cause, and diagnosis of diseased tissue in the brain and spinal cord. And the answer is A, neuropathology. Neuropathology deals with the nature, cause, and disease and diagnosis of diseased tissue in the brain and spinal cord. Okay. Kendra, you still in the first place, but Alyssa, you moved up. Kiana, Angie, Angela, and then Julie. Okay, next, 18. Participates in the coordination of skilled movements and regulation of equilibrium. A, cerebrum. B, cerebellum. C, spinal cord. D, brainstem. Participates in skilled movements and regulation of equilibrium. Cerebrum, cerebellum, spinal cord, brainstem. The answer is B, cerebellum. The cerebellum participates in the coordination of skilled movements and regulation of equilibrium. Okay. All right. Same order, but Rachel, you have moved into the top 5. Okay, next. Number 19. Regulates reflexes and links the body with the central nervous system. A, cerebrum. B, cerebellum. C, spinal cord. D, brainstem. Regulates reflexes and links the body with the CNS. Cerebrum, cerebellum, spinal cord, or brainstem. And the answer is the spinal cord. The spinal cord regulates reflexes and links the body with the central nervous system. Last but not least, Kendra, you're still in the top. Alyssa, Julie, Lakin, and then Angela. Next, movement that turns the palm upwards. Movement that turns the palm upwards. A, flexion. B, pronation. C, supination. D, abduction. Movement that turns the palm upward. Flexion, pronation, supination, or abduction. And the answer is C, supination. This is movement that turns the palm upward. Supination. So, according to this, but I don't know how true it is, Kendra got 14 out of 20. Julie got 13 out of 20. And Lakin got 12 out of 20. So the grand winner, according to this screen, is Kendra. Yay! Okay. Yeah, it's microglia. Great job. But now, I asked you guys to keep a running total of the number that you got correct. So, with that being said, did anybody get 20 out of 20? If so, indicate by saying yes or typing yes. Okay, 19 out of 20. 18 out of 20. 17 out of 20. Okay, 16 out of 20. All right, 15 out of 20. And we know that, oh, nobody got 15. Okay, all right. 14 out of 20. Anybody besides Kendra got 14 out of 20? Kendra, you got 14. If you kept a running total, only those? Okay. You got 14. 13 out of 20. Okay, Amanda, Hannah, Sierra, Brittany, Julie. Outstanding. Okay. All right, so Julie, 13 out of 20. So it didn't record everybody else, but it might have recorded based on time. All right, 12 out of 20. How many people got 12 out of 20? Okay, Lakin got 12 out of 20. Anybody else? Angela got 12 out of 20. Good. Natalie, good. Okay, Kiana, good. Anybody else? Rachel, I think you did too. Okay. Allie? Okay, good. All right, 11 out of 20. Shelton, good. All right, 11 out of 20. Anybody else? 10 out of 20? Okay. Jacqueline, Lindsay, Michelle, Adriana, Tiffany, Chesley, Nicole, Joshua. Good. Okay, 9 out of 20. Jacqueline Wagner, 9 out of 20. Anybody else? 8 out of 20. Not sure what I got. Okay, Rolla. Anybody got 7 out of 20? 6 out of 20? 5? 4? Forgot to count. 3? 2? 1. Okay, guys, so the test that you just had, the little quiz that you had, and I am so sorry that I won't be able, I guess, to use Kahoots because it's so much fun, but I don't know how to show it I can't show it in Blackboard Collaborate, I don't think. I tried, but I couldn't log into it through Blackboard Collaborate. But you figured it out. Did you like that? So what I can do is still do something like this in a quiz. And I can make, I could do that for you. Okay. All right. So if you thought it was helpful, I'm trying to think of ways for us to be interactive. I hate that because I love Kahoots and it has music to it. It is so much fun. Okay. So, that being said, this is recorded for you. You can go back and listen to the questions and the answers. This is how your test will be. It's going to be multiple choice, just like that. You'll have a question, and then you'll have an answer. Yes. Okay, so if you look on page, let me see where it is. There are terms. Remember that in Chapter 1, we talked about terminology that is central to understanding the neuronal structures of the nervous system. And one of those terms is a nucleus. And the nucleus, these are terms that if you look on page 16 of your textbook, and always have your textbook in class, okay, because I'm going to have to refer you to tables. But if you look on page 16 of your textbook, you will see terms that you use when we're talking about the central nervous system versus terms that we use when we're talking about the peripheral nervous system. So if we say nucleus, we are referring to the gray matter, okay? And so a mass of neurons that are usually deep in the brain is a nucleus. And so if you look on page, we have the cerebrum, which is the cerebral cortex, which is the outside of the brain, but then we also have structures that are deep in the brain. If those structures are gray matter, which means that that's the cell body of the neuron, then in the central nervous system, they are referred to as nucleus. Okay, so that being said, if you will turn to page 52, and everybody let me know when you have page 52. Let me show you nuclei. Let me know. Okay, look at figure 215. And I look at the caudate nucleus, the head of the caudate nucleus. Now, it is right below the ventricles. The ventricle are the open spaces. Everybody tell me when you see the head of the caudate nucleus. The head of the caudate nucleus is a nucleus. It's gray matter, okay? Everybody, identify the putamen. Identify the putamen and the globus pallidus. Let me know when you see it. Okay, all of these are part of basal ganglia structures, and they are nuclei. So the caudate nucleus is a nucleus. It is gray matter. The putamen is a nucleus. All of the gray matter that you see are nuclei. Yes. Even though they're basal ganglia structures. Yes, lady.
Okay, so I'm looking at that picture, and the globus pallidus, it's kind of like, there's like a little white streak that's running through. Is that like part of it? Or?
That's not the globus pallidus. That's the internal capsule. If it's a white streak, it's an axon. All the white areas that you see are axons. All the gray areas that you see are nuclei, or they are masses or bodies of deep tissue, or they're nuclei, or they're gray matter, like cell bodies of the dendrite. So where you see the internal capsule, that's white matter or an axon. But where you see the globus pallidus, it is look at the putamen and look at the globus pallidus. It looks like a lens shape, or it looks like a triangle, does it not? Now, if you agree with me in what it looks like, the globus pallidus is the part where at the top of where you see, it's the front part of the putamen is the globus pallidus. And I'll show it to you in another slide. But nevertheless, it's gray matter. And if it's gray matter, it's the cell body of a neuron or a nerve cell, okay? If it's white matter, it's the axon part. So what you have is the dendrites, and then you have the cell body or the soma. So what you're looking at are the nuclei, okay? Everybody got that termed? Everybody understand? And now turn back to page, let's look at the outside. Look at turn to page 25. Yes, nuclei are parts of the central nervous system. That is correct. And if you look at your chart on page 16, table 1-7, but look on page 25 first while I'm making a point. Everybody tell me when you got page 25. Okay. look at the very top where it says anatomic orientation, left, right, the very first picture of the cerebral cortex. Everybody see the first picture? It says left, right. That's the outside of the brain, right? Everybody agrees that's the cerebral cortex? Okay, that's gray matter. That is gray matter. That's the nuclei of the soma of the axon. I mean, not of the neuron. Okay, so the outside is always gray matter, and then you have all this white matter going through. Turn back to page 52. The picture you were just looking at, figure 215, where you have the basal ganglia structures, the putamen, the globus pallidus, and the caudate nucleus. Okay. Everybody let me know when you're on page 52 again. This is point of reference for you. From the perspective of that picture, look at the outline of the cerebral cortex. And the color of it is gray, is it not? And then you got those nuclei that are deep subcortical, the basal ganglia. Now, right before the outline of the brain, right under the outline, you see white matter coming off of the cerebrum. That white matter, the white matter is axon. Those are axons. And what you see is the corona radiata of those projection fibers coming from right where the outline at the top of the brain, the frontal lobe is. You see white matter projecting down. And then where you see the caudate nucleus, in between the caudate nucleus and the putamen is the internal capsule. So those projection fibers from the top of the cerebral cortex projects down to become the internal capsule. Now, you also see fibers that connect the right side of the brain to the left side of the brain, and those are commissural fibers. But the term nucleus refers to the putamen, the caudate nucleus, so forth and so on. By definition, this is a mass of neurons that are usually deep in the brain. For example, page 16, table 1, the caudate nucleus. Lateral genicular nucleus, that's part of the thalamus. And if you see the thalamus on page 52, all that gray matter, all those things are nuclei. Everybody understand? Does everybody understand what nucleus is? It's just deep. It's just a mass of neurons that are deep within the brain. And they can be identified because they're gray matter. Any questions about that? Okay, also look at table 1.7 because you have to know these terms. In fact, the tables that you need to know for your, first of all, you got to know the principles, but let me point you to tables. Okay? First of all, go back to table 1.7 on page 16. Yes, that is to clarify, Jacqueline. All of those are neurons. The body, the brain particularly, is made up of billions of neurons. And neurons have various parts. And we've been talking about the parts, the dendrite, the cell body, the soma, and the axons. The axons are always white matter. And then you have and the dendrites too, and then you have the cell bodies, which is always the gray matter. Okay, everybody got table 1.7 on page 16? Okay, that being said, you need to know these terms. The word nucleus, the word tract, and the word fasciculus. Those three words, when you hear them, they are related to structures in the central nervous system. The nucleus is the mass, or nuclei for plural. So we're talking about mass of neurons usually deep in the brain, like the caudate nucleus that you just saw, on page 52. Tract is a bundle of parallel axons with a common point. So a tract, axons, that's got to be white matter. And if the axons originate in the at the area of the cerebral cortex, and then they travel down, that white matter starts in the cerebral cortex, like on page 52, where we see white matter just below the gray matter, which outlines the brain. The projection fibers then turn into the internal capsule. Those fibers continue to run through the brainstem, and if they run to the spinal cord, then we call that a tract. The beginning of the tract will be outlined by its name. So, for example, if it says cortical, that's where the tract begins. If it says spinal, that's where the tract ends. But it's a bundle of fibers that run parallel to each other. So we have many different tracts. We have the cortical spinal tract, which begins, the axons begin in the cerebral cortex, and they travel down through the spinal cord before exiting through the peripheral nerves to muscles or glands. If it's cortical bulbar tract, B-U-B-L-A-R, then the axons begin at the level of the cortex, and they travel to the brainstem. Brainstem is the bulbar. Another name for the brainstem is bulbar, B-U-L-B-A-R. Okay, so a tract, we said it's a bundle of parallel axons. So I have fibers running from the cortical spinal area down, at the same time that I have fibers running from the cortex to the spinal cord. At the same time that I have fibers running from the cortex to the bulb or the brainstem. So these are parallel groups of axons, so they are called tracts. Any questions? It's not a muscle, it's an axon. A tract is a group of axons that are just running. So think about highways, okay? You have highway, let's say, or think about streets. You have streets that run parallel to each other. They begin at a point and they end at a point, right? Oh, wait a minute. I mean the naming. Oh, yeah, I guess so. Yeah. The insertion of where a muscle is inserted and then where it exits it from that standpoint, yes. Okay, so if you hear cortical spinal, it means that the axon begins at the cortex, ends at the spinal cord. All right, cortical bulbar, the axon begins at the cortex, ends at the brainstem. All right, spinal thalamic, the axon begins at the spinal cord, ends at the thalamus. Okay, so these are tracts. Okay, so a bundle of parallel axons with a common point of origin and termination, cortical spinal. Fasciculus are groups of tracts. Okay, peripheral nervous system is those neurons that are like in the peripheral nerves. The peripheral nerves come off of the spinal cord and the cranial nerves. So, for example, these nerves in the peripheral nervous system, the ganglion, are a collection of neurons. An example would be the trigeminal nerve. The trigeminal nerve comes off of the brainstem. The facial nerve comes off of the brainstem. But ganglion, they may have a dorsal root or a ventral root. Okay? And then if you hear the term nerve, nerve is a bundle of axons, like, for example, the facial nerve, and that's in the peripheral nervous system. So the facial nerve is in the peripheral nervous system. The auditory nerve is in the peripheral. All the cranial nerves are peripheral nerves. All the cranial nerves are peripheral nerves. Any questions about this table? Any questions? Okay. So, that being said, In Chapter 1, absolutely you're responsible for reading the whole chapter. I can ask you anything out of the chapter. But let me tell you how easy it is going to be to study. Look at all the tables in Chapter 1. On page 4, Table 1-1. Go to the tables as I'm talking about it. Okay? You should know the branches of neuroscience. And in Table 1-1, they give you the branches and they give you the definition of the branches. All right. Table 1-2. When we talk about major brain diseases, or the last are neuropathology, Table 1-2 gives you disorder and it describes those disorders. So it's a cerebral vascular accident. It's the sudden loss of sensory, motor, speech, and language functions caused by interrupted blood supply to the brain. But Alzheimer's disease is a progressive degenerative disease of the brain associated with dementia. So they give you definitions of blood problems like cerebral vascular disease, demyelinization problems like multiple sclerosis. That's demyelinization in the central nervous system. They also talk to you about degenerative diseases like Alzheimer's, tic, motor diseases like Huntington's, chorea, Parkinson. So know these, read over these, because you'll have to recognize them. Okay? Look at, make sure you read on page 7 the principles governing the human brain, because Table 1-4 gives you the names of those principles, but you are responsible for understanding what those principles mean so that you can identify them. Okay? So like the plasticity of the brain is the brain's ability to change the result of experience, to change as a result of experience. Okay? Bilateral anatomical symmetry is a principle of laterality of brain organization. That means that the two hemispheres are essentially the same. They kind of mirror each other. Okay? If you look on page 12, Table 1-5 gives you the terms that indicate direction. So caudal, you have rostral, which means near the front of the head, from the perspective of the cerebrum. That's the forehead. Okay, rostral is the forehead. But if you're talking about the brainstem curves. So rostral means towards the brain or near the brain. Know the difference between the directions for cerebrum versus brainstem and spinal cord. Now, caudal means the back of the brain or head, as opposed to dorsal, which means the top of the brain. So make sure you look at your structures. You can look on page 11 to learn those structures, to see the difference. So if you look on page 11 and you look at the brain, you see a dog, and that shows you rostral and caudal, but you see the brain. It shows you rostral, caudal towards the back of the brain, rostral towards the front, dorsal towards the top, ventral towards the bottom. Okay? So, know Table 1-5. Turn to page 14. Know Table 1-6. Okay? And turn to page 16. Know Table 1-7. Table 1-8 on page 18. Okay? Read over those brain structures and be familiar with the functions, because we've talked about the function. Like, for example, the brainstem. If you look at the midbrain, the pons, and the medulla, we're talking about auditory and visual reflexes. We're talking about respiration, phonation, heartbeat, blood pressure, all of those things that are important to life. So your quiz is on Chapter 1, and those tables are primarily, in addition to what you read, make sure you have an understanding of those tables. We are going to finish Chapter 5 tonight. That that being said, 4:40, 4:30, 5:30. Okay, good. Let's look at chapter 5. And what I'm going to show you that you need to know immediately, because then you can go back and listen to this test. You can go back and listen to this test. Okay. So if you look at chapter 5 and you look on page 153, you see figure 5.1. Let me know when you're on 153. Okay. Know the components of figure A, the basic components of the nerve cell and its major parts. So you need to know that a neuron consists of the dendrites, the nucleus, or the cell body. The nucleus is in the cell body. That's also called the soma. The axon helix, the axon, the myelin sheaths around the axon, the nodes of Rumbier. And then you need to know the synapse, that at the end of the axon is a terminal boutons or knobs. You have teledendria coming off of the, which are just branches that come off of the axon, but you have boutons at the end of that. You have a synaptic cleft, and then you have a postsynaptic receptor area, which is attached to the dendrites of the next neuron. You also need to look at figure B and know that within the cell body or soma, you have different organelles, such as the nucleus. The nucleus contains DNA. The nucleolus contains RNA. RNA is responsible for synthesis of proteins. We have ribosomes. We have lysosomes, and rough endoplasmic ribosomes and smooth endoplasmic ribosomes. We have lysosomes. Lysosomes are responsible for doing what? Really getting rid of material that's foreign to the cell, like bacteria and viruses. So it breaks it down and then it gets rid of it. You need to also know that the Golgi apparatus is responsible for packaging proteins, sorting and packaging them to get ready to send them or transport them. And the mitochondria is responsible for energy, and it makes ATP. Okay? So know the components of the nerve cell. So that's nerve cell physiology. The dendrites are receptive. They receive. But the axons are they transmit or they transport, so they're more motor. Okay? Turn to page 155. We talked about the different types of nerve cells, and look at figure 5.2. We have multipolar, bipolar, and unipolar nerve cells. So know that we have 3 different types of nerve cells and be able to describe those nerve cells. Multipolar cells have many dendrites and 1 axon. Okay. Then we have bipolar that have what? One dendrite and one axon. So they have two processes, a dendrite and an axon, one extending from each pole of the body. These are the types of cells that we find in the inner ear and in the retina. And then that unipolar cells are T-shaped. And then they have one process extending from the body and dividing into the axon and the dendrite. And these are sensory in nature. Multipolar are motor in nature. Okay? And then look at page 156. Okay. Oh, nucleus is DNA. Not sure about the RNA one. The RNA one is nucleolus. And the RNA is a messenger. It carries information from the nucleus out into the cytoplasma of the nerve cell. Okay, let me see who raised their hand. If you raised your hand, I don't see your hand. Okay, Rachel.
Which one did you
yes.
say was sensory in nature?
Sensory in nature are going to be your dendrites. They're receptive, they're afferent, or they receive information. The dendrites receive information. So that's why we said they are sensory. So if you look at figure 153 and look at the synaptic cleft, and then look at the postsynaptic receptor area. That's the that's going to receive a neurotransmitter. And the neurotransmitter is either going to excite these dendrites. You see the dendrites, what you're looking at is the dendrite of another neuron in the postsynaptic receptor area. So dendrites receive, so that makes them afferent, A-F-F-E-R-E-N-T. Okay? Okay, but axons are efferent, E-F-F-E-R-E-N-T, because they transmit information from the cell body to other neurons. So a nerve impulse is what goes through the axon, and that nerve impulse is generated by information that's received by the dendrites. The dendrites are either going to be they're going to receive information that's going to either send to the nerve cell, and it will then go from the nerve cell, or the soma, well really the soma, to the axon, or the dendrites will receive information that will inhibit it from responding. And this information is coming in the form of neurotransmitters that can either cause the neuron to fire or to do nothing. Okay? So, let's see. Any other questions? Okay, look at table 5.1 on page 156. And you need to be familiar with the astrocytes, the oligodendrocytes, the microglia, the ependymal cells, and the Schwann cells. And you need to know their locations. So astrocytes, oligodendrocytes, microglia, and the ependymal cells, all of these things, the astrocytes, the oligodendrocytes, the ependymal and microglia are all in the central nervous system, as opposed to the Schwann cells. The Schwann cells are the only cells that are in the peripheral nervous system, and they are responsible for making myelin in the peripheral nervous system. And they are quite different. The peripheral nervous system is quite different from the central nervous system because of the presence of endoneurium. It's a fibrous connective tissue that covers axons. And in the peripheral nervous system, endoneurium only occurs there in the peripheral nervous system. It is never in the central nervous system. That's also why the peripheral nervous system can regenerate it has axons that can regenerate. But that cannot occur in the central nervous system. The power to regenerate does not occur in the central nervous system because psychologically it's very different. The cells are very different. Okay? So that's important to know too. Okay? All right. So the chemical factors that are important for axons to connect with other axons, and the way the axon is developed, I guess is the best way I can say it, in the central nervous system is quite different from what we see in the peripheral nervous system, because the peripheral nervous system has connective tissue, endoneurium, that you will never find in the ever in the central nervous system. And in fact, the fibers in the peripheral nervous system are held together by this connective tissue. So that's the distinctive difference between the peripheral nervous system and the central nervous system. So astrocytes, oligodendrocytes, ependymol, and microglia are found in the central nervous system, but not in the peripheral nervous system. But the peripheral nervous system has Schwann cells, which are never found in the central nervous system. Schwann cells are used, their function is to form myelin sheets around axons in the peripheral nervous system, and they constitute the fibrous connective tissue around fibers in the peripheral nervous system. Microglia, they're going to travel to a site where there's a lesion. So let's say that there's a lack of blood supply to the brain. What happens? The neuron is starving because blood carries oxygen and glycogen, which is what the neuron needs to live. So let's say that the neuron begins to die. Then what's going to happen is the cell body is going to swell. All the organelles are going to swell, and in many cases they may be displaced. The nucleus will be displaced. And as that cell is going through the swelling period, then phagocytosis will occur. And that means that the microglia are going to travel to that spot, and they're going to try to engulf that dead tissue or that damaged tissue. They're going to try to destroy it. And astrocytes, also the blood-brain barrier is going to be broken down, and astrocytes are also going to travel to that area to try to form some kind of lining. Because what happens with neuroglia cells is they want to liquefy that area and kind of destroy and eat it. And so astrocytes then will step in to try to become a lining and to kind of form scarred tissue. So astrocytes are important in two ways. The first is that obviously they kind of contribute to the blood-brain barrier by stopping foreign substances from getting into the blood. But the second way is that they form scarred tissue around dead brain tissue. So in many cases you may have like some cavity there. The astrocytes will try to fill the hole, okay? But the microglia are just going to try to eat and get rid of that area of tissue that is no longer functioning appropriately. Any questions about the difference between astrocytes and microglia? They are all both important in the process of trying to clean up the dead debris within the nerve. But microglia really are the ones that are going to engulf and kind of eat or get rid of the lesion, and astrocytes are going to try to help by forming scarred tissue. And then they're going to try to keep bad things from coming into the bloodstream. Oligodendrocytes form myelin sheets around axons in the central nervous system, and ependymal cells form a lining around the ventricles. So those are the different types of cells that we're going to see, and those are neuroglia cells. So we got two types of cells that we're talking about. We've got neurons, and then we have neuroglia cells, okay? Any questions about those? Because, yeah. That's it, Rachel. The only neuroglia cells in the PNS are the Schwann cells, and they create. And so when you have problems with the peripheral nervous system, well, we're going to talk about that. But if you have a degeneration of the Schwann cells in the peripheral nervous system, that causes a, do I want to say disorder? Well, yeah, it causes a disease called Guillain-Barré syndrome. And what happens is it's an autoimmune syndrome of demyelinization in the peripheral nervous system caused by, usually by some foodborne pathogen. And so what happens is then you end up not being able to feel in your lower limbs when you have Guillain-Barré syndrome. So your lower limbs usually are affected first. Your legs, your toes, they become numb. It travels from your lower limb up to the upper part of your body. Your deep reflexes are associated. Respiration could be associated, so you might end up dying. As opposed to the central nervous system where you have oligodendrocytes. Oligodendrocytes form the myelin sheath in the central nervous system. However, if demyelinization occurs in the central nervous system, it means that you're going to have multiple sclerosis, and that means that the myelin is degenerating. It's breaking down. So it's going to affect the speed of conduction. So therefore, the sensation that you feel is going to be traveling very slowly in your system, and therefore you're going to have problems with movement. And you're going to have problems with your eyes. You're going to have problems, oh my God, all kinds of problems. So we're going to talk about those types of problems that occur as a result of problems with demyelinization in the central nervous system versus demyelinization in the peripheral nervous system. Also, you can have tumors that occur in those areas. You can have astrocytomas, which means that there's a tumor of the astrocytes, and this is the most malignant form of tumor that you can have in the brain. It invades the brain rapidly, and it travels extensively throughout the brain in adults, and it's deadly. You can have oligodendrogliomas, which are tumors that arise of the cells that we're talking about in the brain. Does anybody else have any other questions about what we've talked about thus far? Because we need to talk about, we've talked about the components of the neuron, or the nerve cell structure. We've talked about the neuroglia cells, the ones that occur in the central nervous system, versus the one type of cell in the peripheral nervous system. So what we want to talk about now is what is a nerve impulse and how does it happen? Okay? Oh, okay. Thank you, Mom. Okay, I can't do it right now. Okay, guys, so neuroglia cells are responsible for absolutely. Neuroglia cells are responsible for protecting the brain. Okay? All right, so when you talk about muscular dystrophy, you have a problem with the muscle becoming flaccid. All right, and so we're going to talk about all of these different types of problems that we see. And what I want you to do is let me get through the nerve impulse and then talk to you about the types of problems you have when you have problems with the axon. And then we'll talk about just clinical concerns for this chapter. Okay? Let's see. Let me see. Did I answer the question? Let's see. So only the neuroglia and the yes, the Schwann cells are the only neuroglia in the peripheral nervous system, but all the other ones are in the central nervous system. Muscular dystrophy, it's when the muscle becomes flaccid because the muscle is going to degenerate. And so what we're talking about is atrophy of the muscles and muscular dystrophy. This is an inherited disorder, but we also have myasthenia gravis, and the problem is we have antibodies that are going to adhere to acetylcholine receptors. Acetylcholine is very important for muscle activity, so your ability to contract your muscle, so contract and to extend, to flex or contract, depends on whether or not the acetylcholine is functioning properly at the area where at the neuromuscular junction. And what happens is you have antibodies in myasthenia gravis that breaks down the acetylcholine, where it is going to try to bind to the acetylcholine receptor of the dendrite at the neuromuscular, where the nerve and the muscle comes together. So without acetylcholine, the muscle activity is lessened, so it's harder for you to contract or to to flex or contract. So hopefully that helps. I'll show you. We'll have some. Oh, by the way, you wanted websites? Listen, in the PowerPoint that we have, I tell you to go to YouTube videos, and I give you the names of them. So what you do is go to YouTube and type in the names of the videos, and then just click them on. I have the exact names in there, so you should be able to view the videos, okay? I meant to tell you guys that. Okay. Any other questions? So, what time is it? Do I need to give you a break? All right, I'm on Colorado time, so it's so confusing to me. So if it's 6:09 here, then it's 7:09 Central and 8:09 Eastern. So that being said, I'm normally in class from 5:30 to 9:30, but I'm going from 4:30 to 8:30. So what about let's take like a 15-minute break? Thank you so much, Angela. Y'all help me out. I'm so confused. I'm so sorry. But if we can take a 15-minute break, yeah, keep track, Angela. Help me out. And my people in Colorado, help me keep track. I think I'm supposed to end at am I supposed to end at 7:30 Central? 7:30 Central. 8:30 Central? 8:30 Central. Thank you so much. I am so confused. Thank you, Tiffany. Okay. So what time is it here? Let me look again. It's 10:30. It's 10:30? Okay. Oh, guys. So it's 6:10 now. All right. I'm so confused. Let's take a 15-minute break. Is 15 minutes enough of a break for you guys? I meant to ask that. I got so much to talk about. Is 15 minutes, 15 is 20? Okay, let's take a break. Between 15 and 20 minutes, be back. So if it's 6:10, be back no later than Yes, that is more than we're used to getting for the most part. Okay, then be back by 6:10, 50. Be back between 6:25 and 6:30. Okay? And go to the bathroom, get something to eat, and come back. I'm going to run downstairs and say hi to my brother. I'm upstairs in his home. And grab something to drink. Okay, so take a break right quick, and then we will finish chapter 5. Okay. All right, class, I'm back. Are you back? Okay, good. Thank you so much. I was down there eating. So, let's talk about the nerve impulse. So that's a beautiful picture there of a typical structure of the nerve. But what I want to show you is, and what we're going to be talking about when we talk about the nerve impulse is, let's see if I can get my pencil to work. Okay. Is ions that move, across the membrane to cause electrical charges to change from either positive to negative or negative to positive. That being said, I'll get my right color. So what we're going to be talking about is how does a nerve impulse occur? And generally, dendrites receive, these are dendrites, and I'm just drawing, I'm using blue. Can you see the blue color? If you can see it, let me know. Okay. So dendrites are going to receive some type of signal. It could be somebody touching you. You can feel when somebody touches you or somebody grabs your arm and they're grabbing it really hard. So that's a sensation of some type that may cause you to move your arm or pull your arm away. Well, that sensation, if it's strong enough, it's going to be excitable. And so it will cause an action potential or a nerve impulse. So we're going to talk about the fact that that impulse may flow from a dendrite into the cell body and then into the axon area. Now remember that dendrites are afferent or sensory. They receive information and then they send that information through the cell body to the neuron, the axon, which is efferent because it causes some type of response to occur. On the outside of this axon, you're going to have positive charges. So I'm trying to show, I could probably do it this way. Let me see whether I can do it. Well, let's just draw them. I'm not going to draw all the way down, but on the outside of the axon membrane, you're going to have positive sodium and negative chloride. On the inside, you're going to have positive potassium, and I'm trying to draw negative. So I'll show you this in another slide, but you're going to have positive potassium, and then you're going to have negative proteins in there. And an action potential occurs, or a nerve impulse occurs, when these positive sodium ions actually go inside of the axon and cause the axon to become now positive and the outside of the axon to become negative. So when that happens, that's what we call depolarization, and that means that the signal is being sent down the axon to the axon terminal, and from the axon terminal in this axon bouton, you're going to have a neurotransmitter that's going to jump across the synaptic cleft and go to the other axons. So I'm just trying to draw axons here. If it's excitable, that impulse will continue to travel through the dendrites to the next nerve cell to the axon, and it'll keep going until you know, whatever happens, like the movement that you're going to make occurs. More than one neurotransmitter can occur from one axon, can be sent from one axon, so that's important to say too. So what we're going to talk about is the typical nerve impulse. And a nerve impulse is what happens sensory to make some kind of motor action to occur. Okay? Sometimes the impulse will not be strong, and if the impulse is not strong, nothing will occur. Okay, like something can land on my shoulder and I might not even know it's there. So because I don't know it's there, there may not be any activity that occurs for me to move a muscle or whatever. I simply don't know. There could be a bug or something on my back, and you might say, oh, let me get that off of you, and then you hit it off of me. But what's happening is that whatever that sensation is of that bug being there, I just simply don't feel it, so I don't react to it. Then there could be excitatory nerve impulse, excitatory types of behavior where something touches me and I immediately jump. I jump because it scares me or because it causes a nerve impulse to fire. So does everybody understand what I mean when I talk about nerve impulse, what I'm talking about? Okay, so that being said, all right, let me then see if I can move my slide. Okay, all right. So I'm going to go, we already talked about the different types of glia cells. So make sure you know that information. Remember that, just remember too that an oligodendrocyte, one oligodendrocyte can form multiple myelin sheets, can form myelin sheets around multiple axons at the same time, while only in a Schwann, in the peripheral nervous cell, it's one Schwann cell to one axon in terms of myelin. Okay? Talked about regrowth of Epidermal. We talked about that. Okay. We talked about and saw a video about the different organelles. So make sure you go back and watch that video. Thank you. I'm trying to get to it. Well, I believe I showed you these slides like just by going through the video on YouTube, and that's what I want you to, you can read these slides, but this is what I want you to pretty much function on in terms of learning the structures, because the video is easier. Okay. So I'm going to go now to, so remember, these are all the structures that you can see in front of me. All of these structures are organelles within the soma. And review your video, and that should help you with that. I'm trying not to make it too complex, but I want you to go to the slide that says nerve impulse. Okay? And let me know when you get to this slide, or, you know, if you have your PowerPoint, let me know when you get to your PowerPoint. Okay, good. So, nerve cells communicate through each other, with each other, through what we call a nerve impulse. And the nerve impulse has an electrical component, and then it has a chemical component, okay? And the excitability of the nerve depends on the ion channels in the cell's membrane. So the axon has a membrane, and in that membrane we have protein and we have fats. So we have lipids and then we have proteins. We have different type of, and then we, also have some glycogen as well. Okay, but the excitability of the nerve cell depends on the ion channels that are in the nerve membrane. The ions can either be positive or they can be negative. Okay? An action potential results from charged ions moving through the cell's membrane. So if we're talking about an action potential, then we're talking about a membrane that might have been on the inside, that's on the inside negative and on the outside positive, but now the inside becomes positive and the outside becomes negative. So that's an action potential. Nerve impulses activate the release of a neurotransmitter in the presynaptic neuron. So think about the fact that you have an axon, you have a membrane, you have the outside of the membrane, you have an inside of the membrane. Outside of the membrane is always going to be positive, inside will always be negative. If a nerve impulse or something touches you, let's say, and causes you to move your arm, that's because an action potential occurred and ions moved down the membrane. So the membrane inside may be negative, but now it's becoming positive, like in a chain reaction, just like dominoes. You hit one domino and the next domino falls and the next domino falls. So one area of the inside of the membrane now becomes positive, and then it causes the next area of the inside to become positive and the next area to become positive. Meanwhile, the outside of the membrane that is positive now is becoming negative. So think about a domino effect and ions moving down the membrane. And when they get down to the end of the axon, they then release a neurotransmitter. The neurotransmitter can either be inhibitory, which means that nothing is going to happen, or it can be excitatory, which means the movement keeps going to the next neuron, so forth and so on. Okay, the transmitter often causes the adjacent postsynaptic receptors to open an ion channel. So we saw in the picture just a few minutes ago that I had up in the PowerPoint, and if you look on page 153, you have at the end of the first axon, at the end of the axon, you have terminal boutons. So look at that picture, and it's going to release a neurotransmitter. If a nerve impulse travels down the axon, that neurotransmitter is going to either be excitatory or it's going to be inhibitory. If it's excitatory, it's going to bind to the red area, the postsynaptic receptor area. And that receptor, if it's excitatory, will open and allow the impulse to keep on going. If it's inhibitory, that receptor will remain closed and nothing will happen. So the neurotransmitter selectively opens or closes ion channels to control the excitability or inhibition of the interconnecting neuron. Does that make sense? You're going to have some type of, yes, postsynaptic is the dendrites. That is correct, Jacqueline, or Nikki. That's correct. So here's an example of ion channels on the dendrites of the postsynaptic neuron. They are either in this case, all of this area, you see that the receptors are closed. Okay? So let me just point to that so you can see that. The receptor in this case is closed. It's closed there, it's closed there. So nothing can get through. Nothing is going to go to that dendrite to be sent to the cell or the body or the soma to get to the axon on the postsynaptic neuron because the gate is closed. But if the gate is open, as in this case, something can be sent, that neurotransmitter can be sent, and that impulse can continue to go. So here's an example. And these are just different types of receptors because you have different types of stimulation. You can have a stimulation based on temperature, based on touch, so forth and so on. Okay. Now, so let's look at this. Here is the neurotransmitter is either going to be excitatory. If it's excitatory, the receptors will open. If it's inhibitory, the receptors are going to close. So if the response is enough to cause a reaction, like for example, you grab my arm and I pull away, that's the sensation of you touching me hard causes me to pull away, then that's excitatory and that receptor will open. If I barely know you're touching me or it doesn't bother me and it doesn't cause me to respond in any way, then that receptor is inhibitory. Now, what we hope in the basal ganglia area where you saw the caudate nucleus is that because that's an involuntary area, it's involuntary motor movement. So what happens is we hope that the neurochemicals in that area are going to inhibit inappropriate movement like tremors that you see in Parkinson's or like in Huntington's chorea, that person's ability to stand still and his arm not fly up in a rhythmic motion. So some neurotransmitters are inhibitory, they don't cause movement, some are excitatory. If it is excitatory, then the receptor opens and receives the impulse and it continues. If it's inhibitory, then the receptor closes. Does that help, Jenna, to explain? Okay. Anybody else have a question? So here's an example of the outside of the cell here, and here's an example of the inside of the cells, and they're going across the membrane. Yeah, Whitney, go ahead.
So, excuse me. What I've heard you say sounds like it's as a reaction, things can be excited. But my question is, if like just the actual sensation of the touch, is that transmitted in the same way, or is this only transmitted for reaction's sake?
So I'm not sure I understand the question. There are all different types of stimulation that you can receive. So all of the and there are different types of neurotransmitters that can be released from one axon at the same time. Some excitatory, some inhibitory. So I'm not quite sure the sensations are going to be different. There are different types of and there are different types of receptors based on the types of stimulation that is received. So I'm not quite sure what you're asking, so ask it again.
I guess it's just like when you are like, if someone were to touch you but you didn't have a reaction because it didn't bother you or whatever, is that sensation still being transmitted in the same way, just maybe with different receptors?
Yeah, probably. You're asking how the reaction starts from touch. There are different types of touch. So depending on it depends on how your body responds to the touch. like and those go through different tracks too. There are different tracks for different types of touch. There are different tracks for temperature. There's another track for pain. So because there are different tracks, and there are different types of receptors in the skin, then all of the sensations aren't going to be the same, and they're not going to travel in the same tracks. So how you respond is going to be different. And how you respond is also going to be how you feel about something as well. If that makes sense. It's a real complex process that I'm trying to explain very simplistically, which is hard to do. Okay. Yes. So if the impulse doesn't happen, then nothing happens. You're just at rest. But if the impulse happens, it could be an impulse that's not strong enough to cause a reaction. If an impulse, so it doesn't mean that every impulse is going to cause a particular reaction. Oh, it's not necessarily just touch. I'm using touch as an example. And Natalie, the inside of the cell at rest is always negative. At rest, the outside is always positive. Yes, sensations generally come from outside of the body. They're different, but sensations can also occur, like you can also have visceral types of organs. So you can have involuntary types of sensations that occur just because, like, for example, look at the autonomic nervous system. Oh my God, this is such a perfect example. I went to play bid whist this weekend, and I'm a member of a bid whist club, but so we all have couples that host each month. I'm relatively a new member of the club, so I'd never been to this couple's home before. So I put into the GPS directions to the home. I'm scared of heights. So my directions took me from, and I am getting to a point here, my directions took me from my home to the Natchez Trace. The Natchez Trace is elevation, and the roads are very narrow, and then there's a drop, and there are no, they don't have, what do you call the things? In other words, if I drop off of the drop, that like there's a river there, but there's no barrier there to keep me from going off the road and down like a steep drop. Yes, guardrails. Thank you so much. Okay. Guess what happened to me? Nobody touched me. There was nothing voluntary. My autonomic nervous system just went off the charts. So all of a sudden, I had sensations. I was so scared. I was almost in tears. My hands were sweating. I know my heart was racing 1,000 times. It's my height thing. But the problem was there was a drop. Guess what? It was a winding road, and you could not see oncoming traffic. You know, you didn't really have the yellow dividers. You just knew you needed to be on your side. Guess where I was? I was in the center of the road doing 20 miles per hour. Now, did anything touch me to cause that sensation to occur? It was my fear. And the person that was riding with me she realized I was in trouble. And she said, "You're doing a good job. You're doing fine." And I kept saying, I got to get control of my emotions. I got to get control. And I just prayed that nobody came around the corner, because if somebody had come around the corner, I would have had to move back over to the right. And I don't know that I wouldn't have gone off the cliff. I was so scared. Now, it was a gradual incline, but what was gradual to me might be something that you guys could do. When I got off of that road, as soon as I turned on to where there was grass on both sides, it took me a while just to get my system, the parasympathetic system, to work. Now, that's not an outside stimulation, but that's an internal stimulation that occurred. So me gripping the wheel of my car didn't occur. It occurred because of some reaction internally. Okay? So I don't best know how to explain it, but let me explain what the nerve impulse is. So yes, the stimulation doesn't always have to come from the outside. It could be an internal thing that happens. It could be, okay, so I guess the best way to explain it, let's just explain the nerve impulse and then maybe that'll help. Okay, at resting state, the neuron of the cell is not excited and it does not conduct an impulse. Okay, now remember, what did we say an impulse was? Nerve cells have to communicate with one another through impulses. That is how nerve cells communicate. And so they have a chemical component and then they have an electrical component. A nerve cell has an electrical component because it has ions. The ions are either positive or they're negative. Okay. At rest, the outside of the cell is always negative and the inside is positive. But when there is an impulse or an action potential, the inside becomes positive and the outside becomes negative. So at resting state, nothing is exciting the nerve and there is not an impulse that is conducting. At resting state, the positive and negative ions on either side of the membrane is unequal. We call this state polarized. Okay? Now the reason they're unequal is because you've got, and the reason the inside is more negative than the outside is because on the inside we have negative proteins and we have positive potassium. But potassium is permeable to the membrane, so it can leave the membrane. So you constantly have positive ions leaving the membrane. On positive potassium ions. Okay, but you have on the outside positive sodium ions and negative chloride ions. But the sodium is not permeable to the membrane, so it can't just come into the membrane at will. At rest, potassium can leave the membrane and go out just normally. It just does that. It can cross the membrane, but sodium is harder. So but with chloride, which is negative, chloride can cross the membrane too. So you have chloride crossing the membrane, which is negative, and then you have these negative proteins within the membrane, which are negative. So there is more negative inside because potassium, which is positive, is leaving. Okay, on the outside, positive potassium is leaving, and sodium cannot get into or cross the membrane as easily. So you have positive potassium and positive sodium on the outside, which is making it more positive. But on the outside, but you have an unequal distribution of positive and negative ions, although they are similar in distribution, almost equal in distribution, but they're not. They're unequal, but they're not that unequal, like so greatly unequal from each other. So there is a difference between the electrical charges on the inner and outsides of the cell membrane. Okay? The resting membrane potential is typically between -50 and -70 millivolts in relation to the outside of the cell. So this is below 0. Okay. Now, let's move on. So here you can see, this is black, so I'll just make this blue again, if I can. Okay. Okay. So here you can see at resting potential, Nothing is happening. There is no nerve impulse. This is resting potential. It typically is between minus 50 and minus 70 millivolts. Okay? Now let's see what happens when depolarization occurs. There's some type of stimulus. So what happens is now that membrane is going to change. And therefore, the outside is going to become more negative, the inside is going to become more positive. And we're going to see depolarization. This is an action potential that occurred to cause the change in ions from negative to positive on the inside and from positive to negative on the outside. Julie? And if you look on page 159, please look on 159 and it'll show you the picture of a polarized membrane at rest and then what happens when there is depolarization. Go ahead, Julie.
I have a question and it may be silly, I'm not sure, but energy drinks have a lot of sodium, and I know IV bags have a lot of sodium for like when you're dehydrated. Is that related in any way?
To be honest, I have no idea. But write that question down and ask the medical doctor I'm going to bring in at the end of class. Don't forget, because I don't know. I really don't. But thank you for asking it anyway. Okay? Okay. All right. Is everybody on page 159? Let me know if you're there. Okay, look at figure A. Remember, you have nothing happening to the axon. So on the outside it's positive, and on the inside it's negative. Now you can write, if you want to in your book or somewhere, that the inside negative would be K plus, and then you can put A minus, A for the protein minus. On the outside, you can write Na for sodium plus, and you can write Cl for chloride minus. That's at resting potential, and there's unequal distribution of negative and positive ions. But now, we had a stimulus to touch. Some type of sensory information is being sent through a dendrite, in through the cell body, and look at what is happening now. You got this chain reaction, just like a, what do you call it? Oh, what is that type? Domino effect. So now you can see that sodium is coming into this ion, and because sodium is positive and you still got positive potassium, you've got this chain reaction where the outside is becoming what? More positive. I mean, the outside is becoming more negative and the inside is becoming more positive. This is what we call a depolarized membrane. Okay. You can still see that part of the axon is still polarized, but remember, it's a domino effect. So if you look at figure C, what's going to happen is as depolarization continues toward the end of the membrane, repolarization begins at the beginning of the axon membrane by the axon helox. And the reason that occurs is because while the sodium channel is opening to allow sodium in, and the sodium is moving in a chain reaction down the axon, then the potassium channel, the sodium channel is going to close and the potassium channel is going to open to allow potassium, back in, because as sodium moves in, potassium moves out. Why? Because positive ions do not attract positive ions. They repel each other. So positive repels positive. Positive and negatives attract. Okay, if that kind of makes sense. All right, so here, the membrane potential is sustained by an unequal distribution of positively charged sodium and potassium ions and negatively charged chloride ions and proteins across the membrane. The concentration of positive ions is higher on the outside and negative ions are higher on the inside. This is at rest. A typical neuron then has a low internal sodium and chloride concentration because the internal part is going to be, at rest, is going to be what? Potassium and protein. And then, so it's going to have a higher internal potassium concentration. A nerve, okay, yeah. A nerve impulse, an action potential causes ions, causes the sensation to be strong enough to cause the impulse. Okay, so the potential occurs when sodium begins to enter the cell membrane and the millivolts change, and that's what causes the action potential. The millivolts change from -50, somewhere between -50 to -70 to, it goes up to a positive. Okay, and it only takes like a stimulation of, I think, 5 or 10 millivolts to cause this to occur, if that makes sense. Okay, so if we look at this figure, and we'll talk more, and then I'm going to let you see a YouTube video. If you look at this, you see these green squares here. All of this is chloride. No, a nerve impulse can never happen without an action potential. An action potential is a sensation that occurs that causes the nerve impulse to occur. Okay, so it's a change in millivolts that causes the nerve impulse to occur. Okay? Okay. All right, so let's go get through the process, and then we'll go back and talk about it again. So you can see that there's very low sodium concentration. The red inside, very low sodium concentration inside this nerve cell, but a high concentration of potassium. Look at all the sodium on the outside. Now, you see that there's also a high concentration of negative chloride on the outside, but very little concentration of negative chloride on the inside. All of this is during rest. Then you see these number signs, or these purple things. Those represent proteins, and all the proteins are negative. So at rest, this is what you've got: more sodium and chloride on the outside, you've got more potassium and protein on the inside. Okay, and you can also see it here. Here is this, they're just showing you one segment of this axon, and if you look at this, on the outside you have a high concentration of sodium that's positive, and you have a high concentration of negative chloride. Okay, on the inside you have a high concentration of positive potassium, and you have a high concentration of protein. But you also have some sodium on the inside, but very little sodium and very little chloride. Same thing is true on the outside, very little potassium. Okay, and the A stands for protein. It just stands for protein. Okay. What I also want you to see is you will have some diffusion. There is, if you look over here, there's a sodium-potassium pump. Okay? But what's more permeable to the membrane? Potassium can get outside of the cell, can leave the cell's membrane easier than sodium can permeate the cell's membrane. Okay? All right, any questions about what you're looking at right now? Any questions? Okay, so let's look at this and just listen, and then we'll watch a video. The distribution of sodium, okay, Rachel, go ahead.
Is there always a protein involved in the nerve impulse?
Yeah, proteins are always inside the nerve cell, always. And remember, in the cytoplasma, that's where proteins are being what? Made and distributed to their various areas. But protein is always there. It's just that protein is negative, and that helps the inside of the cell, of the axon, to remain what? Negative, because they don't leave. It's hard for them to leave. Okay? Also, negative chloride is coming into the inside, which makes the inside of the cell negative. But remember that potassium is permeable, so it's positive, and it's also leaving the cell, the inside, making the inside of the cell more negative. But yeah, there's always protein involved. Does that make sense? And I'm gonna, yeah, hopefully that helps. Okay, so the distribution of sodium and potassium ions across the cellular membrane is adjusted constantly by the sodium-potassium pump, which pumps sodium out of the cell and potassium into the cell. So you have leak channels. They're active at rest. They're more permeable to potassium than to sodium because it's harder for sodium to cross the membrane. Potassium ions can easily leave the cell interior, but sodium can't. The loss of potassium, positive potassium ions across the membrane creates a negative membrane potential while the cell is at rest because positive potassium is going out. And you still got protein in there that's negative, and then you've got negative chloride that's coming in that's making the cell more negative. So negative chloride ions move into the cell also to maintain the negative potential. Within the cell, non-permeant negative charge ions balance the chloride. So those protein ions, those A, that's represented by A, they're negative and they cannot leave the cell. So they're always negative, but you got chloride that is permeable and it's coming in. It's more permeable. The electron gradient along the cell membrane keeps the external surface positive and the internal surface negative. So here is an example of potassium at resting. Potassium is able to leave. It's able to leave because it can cross the membrane without any problem. But this is at rest. It's not an action potential that has occurred. And so because potassium leaves, so I'll try to write a K here, but I can't draw there if you can see this. But because potassium leaves, and it's negative, and then it's positive, then you got a high concentration of positive sodium on the outside. The outside then is more positive than it is negative. And the inside is negative because you got these negative, other ions in there. And then you also have chloride that comes in there to keep the inside more negative. Okay? So this is, what happens is an action potential occurs because of some stimuli. So what happens is, and it changes the millivolts. And so that causes then a nerve impulse to occur. Okay? So nerve excitability. Excitability is the cell's response to various stimuli. The stimuli can be temperature changes. It could be mechanical changes such as, touch, anything like that. There are different types of stimuli that the nerve impulse can receive. Okay? Action potential is conversion of the cell's response into a nerve impulse. So some kind of stimuli occurs that changes the millivolts that causes that impulse to occur. Depolarization occurs when the cell interior becomes more positive and reaches a threshold. So now you have an action potential. So what you got is these ions, sodium ions that are positive. A channel opens up and allows the sodium to come rushing in. But positive doesn't attract positive, So what happens is if a positive comes in, then the potassium then wants to go out because it's positive too. Okay? So sodium comes in, and then the sodium receptor closes, and then the potassium receptor opens, and then it allows, you know, potassium to go out or whatever, because positives don't attract positive. So depolarization occurs when the cell interior becomes more positive and reaches a threshold. Now an action potential has occurred because the millivolts have changed, and the inside is like a domino effect. It's becoming negative all the way down the axon, while potassium is going out and the outside is becoming more negative while the inside becomes positive. Hyperpolarized, but then potassium doesn't want to stay outside too long. Potassium wants to come back in because that's where it normally is. So in hyperpolarization, in repolarization, but I have hyper here, but we're going to go to re first. In repolarization, potassium then starts coming back in to the cell, so making the cell more positive, and sodium leaves, making the cell more, making the outside more positive when potassium moves back in, and making the inside more negative when sodium moves back out. In hyperpolarization, the cell's interior becomes more negative. Thus, it decreases excitability, so no action potential. So what happens is the cell is going to become negative when sodium comes in, and then you still have these negative you're going to have these negative chloride ions in there, negative protein ions. And when it becomes so negative, just like a roller coaster, you know how at the end of the roller coaster, what happens is it goes down way below the threshold. So in hyperpolarization, just like on a roller coaster, the speed is so fast. When it gets down to the bottom, it slows down, and then it goes back up really slow. It goes back up the hill really slow. When hyperpolarization, the inside becomes so negative that it cannot fire. There is no longer an action potential. Yeah, to a certain extent, Jackie, I think that's correct. But let me get through this and then show you the YouTube video, because that'll help. Okay, so what I want to show you, and then we'll come, well, let's look at this right quick, and then we're going to go to the video. Okay, so, okay. Inhibitory postsynaptic potential. The postsynaptic cell demonstrates hyperpolarization in response to the neurotransmitter. It is so negative that it does not fire, and so the neurochemical, nothing happens, okay? If it's excitatory, the postsynaptic potential, the neurotransmitter causes a depolarization of the membrane potential, and therefore the inside will become positive, the outside will become negative. A threshold for triggering an action potential varies from cell to cell, but it's typically 5 to 10 millivolts. And depolarization of a cell membrane opens specific voltage-gated ions, allowing ions to flow in and out of the cell. I want to show this will be on the quiz, so I want to show the video to you regarding review of the action potential. But for a period following the action potential, the cell is not capable of producing a second action potential. This is called the absolute refractive period. Action potentials are either all or nothing. They either occur or they don't. Either the nerve is excitable, the nerve cell is excited, or it is not. Okay? If the cell reaches threshold, it always fires an action potential, and the action potential will have the same shape and magnitude every time it occurs. So let's look at this first, and then we'll talk about it again. Okay? So we're going to go to to video. The video we're going to watch is Review of the Action Potential. Okay. Let me do this again. Sorry. I didn't do the www. Okay. Okay, so I'm going to type in Review of the Action Potential. Okay, let me know whether you can see this video, please. Can you see this? Okay, so let me stop it. And let me give you the website. I know some of the computers are slower than others, so this is what the web address is. Okay, and I'm going to start it again. Okay. All right. Can you see it now? Okay. Can you hear it? Okay. All right. Listen to him. So you see, it's like a domino effect. This is going to be the repolarization part. Okay, so two points that I want to make as we just finished looking at this video. So remember that a stimulus occurs that causes a change in millivolt. That causes an action potential. But the action potential is the change in millivolts that occur as a result of the thing going from resting state to depolarization, repolarization, hyperpolarization, back to resting state. So it's the whole thing. The action potential causes the nerve impulse to occur. Nerve impulse occurs when there is a change in the electrical state of the membrane of the ion. So the outside that was once negative now becomes more positive. After the inside that was more negative, the outside that was more positive is going to become more negative after the inside that was more negative becomes more positive. So in resting state, nothing happens. The outside of the membrane has more sodium and chloride ions, sodium being positive, chloride being negative, and the inside of the membrane has more negative ions, potassium being positive, and protein being negative. So more potassium and negative proteins on the inside at resting, more sodium and chloride on the outside, with the outside being more positive. A stimulus occurs if it's strong enough, sodium pumps open, sodium runs into or comes into the inside, causing the inside to become more positive. It does it like in a chain reaction. And then what happens is the potassium wants to get away from the sodium because sodium is positive and potassium is positive. So in the repolarization state, hyperpolarization occurs when sodium comes in to make the inside become more positive. Repolarization occurs when potassium goes out of the membrane. It wants to get away from the sodium, so it goes out of the membrane to the outside. Okay? Hyperpolarization occurs because potassium wants to come back And inside, sodium wants to leave and go back outside, but the inside becomes way negative. And when it becomes way negative, it's hyperpolarized, it's too negative. It's way below the resting state, and it can't do anything. Nothing can fire until eventually the membrane comes back to, the inside and outside comes back to unequal distribution of ions on the outside and inside. And so that is the action potential, if that makes sense. So that's what causes you to be able to move your muscles, so forth and so on, or to react in particular ways. It's a nerve impulse. If one neuron cannot communicate with another, you don't have a nerve impulse, and then you have problems. When somebody has a stroke, something dies. The neuron cannot communicate with other neurons because that area is affected, and neurons begin to die. And so therefore, that you no longer have what? Nerve impulses, and that's a problem. Okay? Oh, okay, good. Lillian and then Michelle, I'll answer the question. In order for your neurons, in order for you to do anything, to communicate, to think, to walk, anything, your neurons have to be functioning appropriately. And the way they communicate with each other is through action potentials. And you have electrical changes in the axon, and then at the end of the axon you have a chemical reaction called neurotransmitters. Some neurotransmitters inhibit behavior. Some neurotransmitters are important for movement, like, for example, acetylcholine is a neurotransmitter that can only be released if a nerve impulse occurs. And if for some reason there's a problem receiving acetylcholine by the other neuron or by a muscle, then a muscle can't move. How does this affect you as the SLPs? If neurons are not working properly, if they are destroyed, depending on the area of the brain where the damage is, it can affect your speech, it can affect your language, it can affect your ability to comprehend. So that's why you have to understand. Also, the video we watched before, didn't it say that? Yes. And also, too much of some of the neurochemicals can cause all kinds of problems. Like, let's think about norepinephrine. Okay, norepinephrine. This helps us with maintaining attention, and so if you have if you don't have enough of this, then you're going to have problems with attention. But dopamine, if you have excessive dopamine, it causes depression, but it also contributes to schizophrenia. So these neurochemicals are very important, but how do the neurochemicals get released? They only get released if there's a nerve impulse that occurs. Okay, and so that's why you have to understand. So the reason why you got to understand about the ions is because neurons have an electrical component and they have a chemical component. The electrical component is the exchange of ions during a nerve impulse, and the only way a neuron can communicate with another neuron is through a nerve impulse. Okay? So the stimulation has to be strong enough to cause a nerve impulse. And then once the nerve impulse travels down the axon, when it reaches the end, there's a chemical reaction that occurs. And that's the release of a neurotransmitter that crosses over the synaptic junction or the synaptic cleft, and then it binds to a receptor on the next neuron. where the dendrites are. Now, if it's inhibitory, nothing happens. And that's what we call the hyperpolarized state. It means that the area is so negative. It's like a roller coaster. Coming down you get to the top of the roller coaster, and the roller coaster has to come down. Gravity carries the roller coaster down so fast that it goes well below the point in where it started, like the resting state. And nothing happens. And then all of a sudden, it gradually, slowly goes back up. Okay, that's what happens with these electrical ions. Hyperpolarized state means that the receptor on the next neuron is inhibitory. Nothing can happen. If it's an excitatory neurotransmitter that is released, it means that after it reaches an action potential, it fires again, and the and the nerve impulse continues to travel. Okay? Does that kind of help? So, any questions about this, guys? I know it's a lot to digest, but read that area, look at the video again, look at the figures, and hopefully that'll help. Michelle?
Okay, so I have a couple of things. The first question is, in the video, when he was talking about a stimulant, it could be possibly something you ingest, like caffeine, correct?
Good. There are different types of stimuli, like I said before. It could be chemical. It could be something that you put into your system. But I'm not a biologist, so I don't know about all of the different types of chemical reactions, or things that can stimulate. I know temperature is one. I know anything mechanical outside. I know anything internally that involuntarily happens. So that being said, this is not my area. Is not biology, so I understand the basic physiology of the nerve impulse. Caffeine causes you to become more active. It is a stimulant, so it could be.
Well, I was just trying to think, as he was doing the video, I was just trying to make it make sense to me in my day.
But doesn't there have to be anything that you ingest? It could be something that's okay, let's make it simple to something that you would do every day that would be some type of stimulation. Okay, that occurs that makes you feel something. Because remember, they're tracks. Okay, that there are different tracks running through your, I guess, running, well, I don't even know how to say it, but there are different sensory tracks and there are different motor tracks that cause various things to happen. So how do you feel? What is your reaction to pain? Because pain is a track. So what's your reaction to pain? How do you react? How do you feel pain?
I mean, it usually hurts, and you try to assess where the pain is at.
Okay, so now if it's a pain from an outside
like a
stimulus, it could be a bee sting. So the neuron then is there's going to be a nerve impulse that occurs as a result of that stimulus. It could be a sting. So that neuron has to interpret that sting as in some way so that it gets to your brain. And it's going to get to the parietal lobe. That's where it's going to finally end up being interpreted, in the parietal lobe, because pain is sensory, right? But if one neuron is not communicating with the neuron, you're not going to be able to interpret pain. Okay, so let's look at those people who have problems feeling pain. They don't feel pain because there's a problem with the neuron. There's a problem with the nerve impulse being sent forward so that it can be interpreted in the parietal lobe, if that makes sense to you. So what neurons, in order for you to do anything, your the neurons in your body have to be functioning appropriately. And they all make different types of connections. So your body is made up of billions of neurons. And some of the connections are very specialized. In fact, during the embryonic period, you have more neurons than you need. And what happens is more than 50% of them are going to die. And those that don't die become highly specialized. And then some connections are connected to other neurons because they travel where they are attracted or where they're supposed to be. That's the best way I can say it. And in order for you to function, to move, to feel, to do anything, it's got to be through nerve impulses. So how do nerve impulses occur? You have, if the neuron isn't doing anything, nothing happens. You don't feel anything, no pain, no nothing. You don't move, you don't do anything. I mean, I'm just being like really over simplistic of this. But if, for example, I'm going to feel something or move something, I'm only doing that because a nerve impulse is firing, telling me that what? Okay, I got some type of sensation that's causing me pain, and that's traveling on a track and it's going through my brain. And the only way that's happening is that those neurons are functioning appropriately. Now, let me give you a perfect example. When somebody has a stroke, neurons are damaged in a particular area. If they're damaged in the frontal lobe, nerve impulses are not being sent in the area of Broca's, where Broca's is, and guess what? The person isn't going to be able to speak. When somebody has a stroke, movement is affected, right? The right side affects the left side, but that's because the neurons are working properly. Nerve impulses are occurring, neurochemicals are being released, neurons are communicating with one another, and when I'm supposed to move, I'm able to move. When I want to raise my arm, I can raise it.
People with this, like with these types of diseases that we might be working with, they may never be able to reach that high peak, right? Or they may reach.
That's right. They may never be able. You just said it. And most of the time, what happens in a stroke? Do these people? It depends on the extent of the damage. It depends on whether or not the physician can do some type of treatment to reduce the inflammation and swelling, because we're going to talk about that. We're going to look at what happens to what is the neuronal response to brain injury. Well, swelling is going to occur, but the response of the neuron is to try to repair itself. So the RNA is going to start secreting in response to injury. The RNA is going to try to synthesize more proteins, okay? The cytoplasmic structure is going to increase. This is in an effort to what? To repair the cell. In some instances, if the cell is able to be repaired, then that neuron becomes normal again. In other instances, if it's too late, because it goes through a process, this repair process goes through periods. And so we're going to look at that, and we're going to talk about that. What happens is if the cell is totally damaged and it cannot be repaired, then the function is gone. You're no longer ever going to have the impulses in that area again, okay? So the nerve impulses occur in relation to some stimuli, but there are different types of stimulants in the environment, okay? So does that kind of help?
Yes, it does, and I appreciate it.
In answer to Rolla, a study guide, I can't give you a study guide other than what you have. I'm telling you what to read. Like, for example, in Chapter 1, I told you to study Table 1, 1, 1, 2, 1, 5. You got to read, and you got to remember this information. You got to do the same thing with your slides. I can't give you. The answers are in those tables, and they're in the PowerPoints that you're looking at. It's up to you to learn the information. So I don't know what guide to give you other than me pointing out to you what you need to know. And you just got to learn it. So it's hard for me to give a study guide. The PowerPoints are a study guide. Hi, can you hear me? Can you hear me? Okay, yes. So in answer to a study guide, you have to study your tables. You have to study your PowerPoints. You really have a study guide. I can't give you any more of a guide than what you already have, if that makes sense. You just got to learn it, okay? And that requires you taking the time to read, and then to go back and and it takes time to grasp these concepts because they're brand new, if that makes sense to you. All right, so remember, your body, in order for you to do anything, and here's your proof. If somebody has a stroke, they have damage to the brain, they have damage to neurons. If those neurons aren't working, they can't talk, they can't walk, they can't think sometimes. If there's problems with the neurons, if they have tangles in them, if there's a problem with the neuron, they are not able to function normally. If they're functioning normally, then the neurons are fine. Then when there's a nerve impulse that occurs, something happens. That it could be I'm able to talk, I'm able to walk. It could be I'm able to think, because neurons are very specialized. They connect to each other. We lose a lot of our neurons, again, during, you know, when we are going through the embryonic period. We have way more neurons than we need. Those neurons that find connections, those are the neurons that survive. Those neurons that are damaged, those are the neurons that go away. And I'm talking about doing normal development. So 50% or more of the neurons, they're just going to die and go away. That happens normally. But after the neurons that are supposed to be there form their connections so that I can walk and I can see and I can talk and I can think. After those neurons form their connections, when there's damage to the neurons, then you're going to see problems. If there's damage to any part of a neuron, like, for example, if there's damage to the axon, then you're going to have problems. You're going to have problems with movement. You're going to have problems with sensation, so forth and so on. Okay, so I heard somebody raise their hand, but I don't see the raising of the hand. So whoever raised their hand, raise it again. So are lesions caused by, okay, and so thank you, okay, thank you, Nikki. So are lesions always caused by infection or blood clotting? They could be caused by tumors. Lesions can be caused by somebody shooting you in the head, a blow to the head. They could be caused by disease. They could be caused by lots of things. Okay, so you got tumors that could occur. You got those are new growths or things like that. Does that kind of answer your question? They're not always caused by infection or blood clot. You got somebody comes up to me and hits me in the head. They hit me hard enough, then they could damage my neurons. Okay, that's what a concussion is. Okay. Somebody shoots me. Your neurons, they're going to damage the cerebrum, and the cerebrum is made up of neurons. And neurons are made up of what? Dendrites, a cell body, axons, terminal boutons, so forth and so on. Okay, so if it's not from blood clotting from the hit, Okay, it doesn't even have to be blood clotting from the head. It could be that you actually destroyed my neurons. It could be from internal bleeding or whatever. It could be from me eating the wrong foods. I like foods that are high in salt. They could cause plaque to occur in my arteries. The blood can cause what? It could cause a problem with blood flow, and as a result of blood flow, I can then have what? Death to the neuron, because the neuron is not getting enough oxygen and not getting enough glycogen. Okay? Yeah, that makes sense. So damage to the neurons can occur in many forms. So let's then look to see what can happen. Okay? But any other questions before I leave this part? Because I'm going to just briefly review the parts of the neuron for you again, and then we're going to talk about the neurotransmitter, and then we're going to talk about injury. Okay, so let's talk about the synapse. Yes, poison, particularly drug poisoning, okay, doing cocaine and illegal drugs or taking too many drugs can cause oxygen not to get to the brain. Oxygen not getting to the brain can cause death of a neuron, okay? Okay. Synapse is the site of contact of where you have one neuron communicating with the other. In between those two neurons, we have a submicroscopic space that's called the synaptic cleft. That cleft exists between the bouton of one neuron and the cell body of another neuron, or it could be between the bouton and the axon, or it could be between the bouton and the dendrite. So synapses, so we can have different types of communications between neurons, not just the terminal bouton with the dendron, but it can also be with another cell body, or it could be axons communicating with axons. Okay. The axon of one neuron may terminate in only a few synapses or many thousands of synapses. So one neuron can terminate in thousands of new synapses. And if we look at this on page 153, you see you got telodendria coming off of these axons. So you could have and you've got boutons at the end of those telodendrians. So we could have thousands of different synapses that occur. A dendrite cell body complex may receive synaptic contacts from many different neurons, up to over 15,000 synapses. So you got these dendrites, you got different types of cells. You got multipolar cells that are motor in nature. You got unipolar cells that are usually found, they're sensory in nature. And then you have bipolar cells that are found in the nose and in the eyes and in the ears. These are all different types of neurons. They, dendrites, if I've got multipolar dendrites, I've got many dendrites in one axon. So those many dendrites can receive, neurochemicals from many different neurons, okay? A cell membrane of the axon at the synapse is a presynaptic membrane. A cell membrane of the dendrite cell body complex muscle or glandular cell is the postsynaptic membrane. So you got a presynaptic membrane, you got a postsynaptic membrane. Neurotransmitters are released at the synaptic junction. This is the chemical component of the neuron that we're talking about, these neurotransmitters. The electrical component are the ions that travel when we have a nerve impulse that travel down the axon. Peripheral nerve fibers consist of an axon surrounded by neurolemma, and this is what we call Schwann cells that produce the myelin, and it is also connective tissue. Those fibers in the neural lemma cells elaborate a myelin covering, and this makes those fibers myelinated. Okay? So if you don't have any myelin, then the fibers are unmyelinated. But myelin sheaths is important because they help with the speed of transmission of the nerve impulse. Okay? And those myelin sheaths are interrupted by the nodes of Ranvier. Yes, this is funny, but that's true, Michelle. Dendrites going everywhere and receiving stimulation from many different neurons. Okay? All right, the distance to one node to the next is an internode. Okay? All right, and the node are the nodes of Ranvier. So if you look at page 153, they point out the nodes of Ranvier. Okay. The diameter of a fiber and the length of an internode are directly related to the speed of conduction of the nerve pulses. So the speed of conduction is faster for some neurons than it is for others. It depends on the type of neuron it is. Okay. So let's see. So here's a neuron synapse. One, you have synthesis of a neurotransmitter and the formulation of vesicles. That occurs up in the cell body. Then we have transport of the neurotransmitter down the axon. So that's where you see number 2. You see those arrows going down the axon. Okay? 3, we have an action potential that goes down the axon. It travels down the axon. The action potential occurs obviously when you have the negative inner part of the axon becoming positive, the outer part of the axon becoming more negative, and then it going back to, it's reversing itself. That whole thing is the action potential. At the end of this action potential, we have these vesicles, and in these vesicles you have neurotransmitters. Okay, now, so here we go. We have this presynaptic terminal. We have an action potential. It releases these neurotransmitters that jump over and bind to a receptacle. So this is where you see me drawing the blue. Okay? All right. The neurotransmitter is going to either attach to the receptor, and it's going to either excite the receptor, meaning that the impulse then will continue on to the next neuron, or it's going to inhibit. And if it inhibits, then it's in its hyperpolarized state. It's too negative to fire, and it's not going to fire. Then you're going to have a separation of the neurotransmitter. This is step 6. And in 7, some of these neurotransmitters will be sent right back up the axon. They'll go right back on up to the cell body to be reused again. Okay, so here that's step 8. You see the neurotransmitters going right back on up into the cell body to be used again. Some of them will be destroyed, some of them will be reused. So this is a neuron synapse. Okay, any question about that? All right, so I know it's a lot. Okay, so let's look at how neurotransmission works. So here's a new YouTube video on how neurotransmission works. Okay? Yes, an example of a neurotransmitter, and we're going to talk about it, but there are many different types, but we'll talk about them tonight. There are, there's dopamine, that's a neurotransmitter, serotonin, norepinephrine, acetylcholine, all those are neurotransmitters. Aminobutyric acid, glutamate, then you have peptides. You have small molecules and you have large molecules. But let's look at how neurotransmission works. Okay. So I'm going back to YouTube. Okay. Okay, this is quick, fast. This is quick and fast, okay? How neurotransmission works. Tell me if you can see the video. Let me stop it. Okay. Okay, what I'm going to do is start it over again. Okay, I'm going to back it up, and we're going to look at it from the beginning. Does everybody see this? Do you see it? Okay, I'm going to play it. This is neurotransmitters being released. That's if it's excitatory. And this is the dendrite where the receptors are. Okay, hopefully that helped. Okay, good, I'm glad. Okay, yes, Michelle.
Cleft is less than 40
Can you hear me
nanometers
over
wide.
this video?
By comparison, a human hair
I can.
is
Okay.
1,000 nanometers wide.
So I didn't realize
The neuron where
that
the signal is
if
initiated
there were
is called
any
the presynaptic
extra
neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are
neurons
simple
that were
signals
sent
called
off
neurotransmitters
that they
that are
actually,
packaged
if they
in
weren't
small sacks called
absorbed, they were
vesicles.
actually sent back.
Each vesicle can contain
And
I
kind of
can't hear you.
like-
Okay, go ahead.
When
Can you
the
hear me now?
presynaptic neuron is excited by an electrical signal called an action potential,
Hang on.
this causes the vesicles to fuse with the presynaptic membrane and release their contents.
Can you hear me now? Yeah,
Michelle?
can you hear me now? Yes? Can you hear me? I think the video stalled. Can you hear me now?
Okay, while you're listening to that video, when you come back on, you can ask the question. And then I'm just going to move us forward, and then you can come back and ask your question, Michelle. Because I need to get through this lesson before the end of the night because you guys have a quiz on it. So let me know when you're ready, okay?
Okay, can you hear me now? Okay, so it seems like, all right, so I didn't realize,
Yeah.
so I didn't realize that when
Okay, say
neurons
it
were
again.
being sent off,
Say it again.
those that were not, Yeah, so I didn't realize that when the neurons...
Okay, yes, I can hear you. Go ahead.
You can't
what
hear
did you
me
say?
or you cannot?
Okay. I can.
So when the neurons were being sent off...
I can. Go ahead.
The ones that were not being absorbed, they were actually...
I can. Yes, can you hear me?
So were the neurons that weren't being absorbed, were they actually being sent back and held until they could be released again?
I can. We can hear you guys. Can you hear her? Okay, I think you have a delay, Michelle. Yes, the answer to that is yes. So what you have is this uptake effect that occurs. Some of the neurons will be absorbed, but the other ones will be sent back in and repackaged again to be used again. And so what you saw in the slide that I showed before, if I can go back to the slide, is this this uptake, okay? So I've already written on the slide, so it's kind of hard to show this. But if you look at step 6, well, let's go to step 4. You have an action potential that causes calcium to enter, evoke and release of the neurotransmitter. So the neurotransmitter in step 5 is going to attach to a receptor, and either it's going to be excitatory or inhibitory. And if it's excitatory, the nerve impulse continues. If it's inhibitory, nothing happens. But then what happens in step 6 is you have a separation of the neurotransmitter from the receptors. Some of them will be absorbed where you have in the synaptic cleft, and then others are going to go back to be recycled. So that's step 7. And then what you see in step 8 is those vesicles without neurotransmitters, are transported back to the cell body. But some of those neurotransmitters are going to go right back into those terminal Bouton vesicles where they are where they are kept again, stored again to be used again. Then those vesicles that don't have neurotransmitters are going to go right on back to the cell body where they're going to be repackaged and sent back down again. So if that makes sense, that's the way it kind of works. Okay? And this takes time, guys. You guys are doing a great job. You really are. So hang in there. Hang in there with me. It just requires reading and watching the videos and just internalize. You got to think about it, but you can't just look at it one time. You're not going to learn this unless you spend time doing this every day. Oh, don't be discouraged. It's a lot, but you can get it. I promise you. Remember, I've been doing this a long time, okay? All right, let's look at the different types of neurotransmitters that are being released. We have acetylcholine. Acetylcholine is critical in your cycle for sleep and wakefulness. But the big thing I want you to remember about this, it's important in inhibiting basal ganglia activity. Now, the basal ganglia is supposed to really inhibit inappropriate movements. And so the reason I can talk to you and stand still and talk to you is because the basal ganglia is allowing me not to move inappropriately. Now, I have an Aunt Geneva. My Aunt Geneva is a neighbor in my mother's neighborhood. Not really my aunt. But she had taken some of the drugs that she took for, what was she taking? Botox. She took Botox for spastic dysphonia. Her vocal folds were sticking together, and then it's a glottal attack, so hard release. So the Botox was used to get rid of the inflammation and allows the vocal folds to open and close easily. With that being said, what happened with her problem was that because that's a neuroleptic drug, it caused these inappropriate movements, which meant that she cannot control right now to today this facial movements. When she's chewing, she's opening and closing her mouth, she's grimacing. There's nothing she can do to change that. Acetylcholine regulates that activity. It helps to inhibit inappropriate movements. It is also important in helping you to contract and flex your muscles. So it's important in the peripheral nervous system for regulating muscular activity. Dopamine is another neurotransmitter. It regulates basal ganglia motor function, and if you don't have it, like in somebody with Parkinson's, if it's depleted, then you're going to have inappropriate movements, such as resting tremors. The posture is going to be stooped. The movement is going to be reduced. And dopamine is also important in making you feel good. So that's why when people get high, when they do drugs, it causes dopamine to be released in the brain, and the person feels good and they just want to get high again. But because it causes pleasurable feelings in the prefrontal area of the brain. So that's what's happening when somebody gets high. And what happens is if you have too much dopamine, you have schizophrenia. It causes schizophrenia. If you don't have enough dopamine, it causes you to have inappropriate movements. Okay, so you don't have enough acetylcholine, it causes you to muscle weakness, so you cannot flex or contract your muscle, and it causes problems with sleepfulness and being, sleeping and waking. So these are the different types of neurotransmitters that are released. Norepinephrine is primarily in the peripheral nervous system. It's responsible for fight or flight reactions that we have, like me having the autonomic nervous system go wild and my sympathetic nervous system just going out of whack when I got on Natchez Trace. Okay? So the neurotransmitter that is released in that case is norepinephrine. In the central nervous system, it regulates sleep. It also helps you maintain attention for long periods of time, and it also helps with mood. Serotonin in the central nervous system is in the blood platelets and in the gastrointestinal tract. It's also important for pain perception and your overall level of arousal. Lack of serotonin occurs in someone who has severe depression. They don't have enough serotonin. My husband is a person like that. He has severe depression, and so he has to take medication for that depression. Those people, a lot of times, who commit suicide or have depression problems, you'll find that they don't have enough serotonin, and so and the serotonin is being depleted, so the medications allow them to have more serotonin and to regulate their activity. So my husband has to be on medication. His father was severely depressed, and that was part of the problem, and he eventually ended up committing suicide. So these neurochemicals are very important to the way we function and how we function. And so you asked about the importance of neurons and nerve impulses. In order for a nerve impulse to occur, what has to happen? There has to be some type of stimulation. And once the nerve impulse gets down to the end of the axon, there's going to be a release of a neurochemical. And it could be any of these neurochemicals that we're talking about. GABA is a derivative of glutamate. Its process is to inhibit inappropriate movements in the central nervous system. It's a major inhibitory neurotransmitter. It also regulates pain perception. So somebody with Huntington's chorea. Huntington's chorea is a problem with the caudate nucleus, which we saw tonight. The caudate nucleus is part of the basal ganglia. It's right below, it's one of the nuclei that we talked about, right below the ventricle. And those people with Huntington's chorea don't have enough of glutamate. What happens is it causes them to have this inappropriate movement, this dance-like movement, and they can't control it. Okay, and then we also have glutamate, which is an excitatory neurotransmitter in the central nervous system. And finally, all of these types of neurons, neurotransmitters are, they're a subgroup of small molecule transmitters, but the larger molecule transmitters are things that we call peptides. An example of a peptide is endorphins. So you know how when you exercise you feel really good. The more you exercise, the more you want to do it and you feel good. That's because this large molecule called endorphins, they are released. And so that's what peptides do. So what I want you to do is, if you look at table 5.4, you will see, and it's on page 166, you will see the neurotransmitters, you'll see the functions. So make sure you review those, okay? You don't have to. So what you need to know is, for example, acetylcholine is responsible for muscle contractions and muscle activities, things like that. Lack of dopamine causes severe depression, but it could also lead to schizophrenia if you have too much dopamine. Okay? Let's see. Let's talk about neuronal response to injury. You don't have to know cytosecretion, but what you do need to know is what the function of these, you do have to know the function of the neurotransmitters. Yes, Michelle?
You need to get through this lecture.
I'm not going to rush through it.
Okay.
Now I'm only going to go as far as I can go.
All right, so here's something that just kind of red flagged.
So go ahead. You're always welcome to ask a question.
This table that you had us turn to. Dopamine, it's a pretty...
Hold on for a minute.
Okay, well, dopamine is just, it's a pretty common thing, and I think a lot of us know a lot about it. But looking at this table and reading what it says in the table doesn't say anything about the comment that you just made, which I agree with you on all of that, and really we've all kind of heard that. That's a problem that I kind of find with tests.
Okay, but remember I said that you must also read the chapters. You must also know your PowerPoints. So it's not just knowing the tables. The tables try to give you a review. But in order for you to understand it, you must read. You are not excused from reading. Everybody has to understand that. And the PowerPoints actually pull out the information from the readings. Mm-hmm. So if you were to go and read the section on dopamine, you come down into your second paragraph, and down at the bottom, it tells you excessive dopamine activity in the forebrain contributes to schizophrenia. So what I've done in the PowerPoint so that you guys will understand is I've taken the information actually from the book, from what you should be reading,
yeah, and I will
pulled
say
out the important parts and put it in the PowerPoint.
Slides have been excellent in comparison with reading it. Yes, it's so hard.
Because reading it is hard, but I need you to read it, but I need you to review your PowerPoints and the tables as well, because a lot of time in the quizzes, what I will do is I will take the wording exactly from the table or the book. So it's not like in the quiz that you had today, that you guys were doing. That's just the wording from the book. But what I did was to simplify it. So the best way to study would be to review the table and to read the PowerPoints, because the PowerPoints are not my words. All I'm doing is taking it from the book and trying to pull out the important parts. So you can do, if you study your PowerPoint, you still got to read. You are not excused from reading. But the point is that in order to understand it, like for example, look at the slide in front of you. Neuronal response to injury. That's coming from the book. But I'm just making it simpler for you. Because, I mean, you know, but if you read it, you might understand it better. Because, see, for me, I can just visualize it and see it so clearly, but why can I do it? I've been doing this for a long time.
I read it, but I don't really understand it until after I hear what you have to say, and then I go back and reread what your PowerPoint say. So it really does, I think your PowerPoints really complement what the book has to say.
Yeah, so the book I want you to read, but focus on the table. When I tell you to focus on a table, what that does mean is that, like, I'm going to probably use that wording in the table, but not necessarily for this dopamine stuff or whatever. Because what I really want you to know is what? Okay, if it's too much of it, schizophrenia, not enough of it, severe depression. That's what the slide says. But use the slide, but you still need to read the book because maybe my slides aren't enough to help you understand, if that makes sense. Okay? Do you ask case? No, I'm not asking case study questions on the exam, but we will talk about the case studies. Okay? Yeah. All right. Let me explain. I want to explain to you neuronal injury, and then because I want you to, you'll see myasthenia gravis, you will see multiple sclerosis, and you will see Guillain-Barré syndrome on your quiz. Let me get through those right quick, and then we will be finished for the evening. But I also have 3 YouTube videos that will help you. Okay? So very quickly, you have neuronal response to brain injuries. Look on page 161. Okay? Now, remember we said that a neuron can be injured for various reasons. Could be because of a tumor, could be because of some type of bacteria, a virus, could be because of a blood supply problem. Okay? But what happens is the neuron may degenerate. And if it degenerates, it means that what? It is either going to degenerate in the presynaptic area or at the postsynaptic terminal. So we have 2 types of degenerative changes that occur if there's a problem with the axon. Okay? Now we're talking about a problem with the axon. And it could be axonal retrograde reactions or Wallerian axonal reactions. Now, if it's axonal retrograde, look at figure A on 161. And that means that, now you can see where this axon is broken into. Everybody is on page 161? Please get on 161. Because I need you to understand this. It is not a hard concept, but you need to understand it. Okay. In figure A, what you see is there's an injury to this axon, and where you see the red is where the injury has occurred. And you also see that, so there's a proximal injury to the cell body. Proximal means closer to the cell body, okay? Then you have a distal part of the axon, and you can see that that distal part is broken away. So what we have is axonal retrograde reaction, and then we have Wallerian degeneration. And there's the lesion. The lesion cuts the axon in two. Now, let me explain this. If it's axonal and it's retrograde, then this means that the retrograde degeneration occur in the cell body or the soma. In other words, the cell body is going to react to the injury that occurs in the axon that's closest to the cell body. So the organelles in the cell body are going to start to degenerate. They're going to swell. When they swell, that means like the mitochondria begins to swell, the lysosomes, the Nissl bodies, all those things. The Nissl bodies, in fact, they begin to kind of break down. So just think about the organelles reacting to this problem with the axon, and they swell. The blood-brain barrier is altered. So that means that bacteria and viruses can get in the blood because of this problem with the axon. The nucleus in the cell body is going to be displaced. Normally, the nucleus is in the middle of the cell body, but it's displaced, so it moves more to the side or to the peripheral area. And that's what we call chromatolysis, where you have the swelling and you have this displacement of the nucleus. Okay? Now, you could have, the cell could die, but remember that the RNA synthesizes proteins. That's what it does. So the RNA wants the cell to recover. So cellular RNA production and protein synthesis is going to increase in an effort to try to save the cell. The physician hopefully is going to step in and do some type of medical treatment to try to help in the reduction of what's going on with the neuron cell. The swelling is going to decrease on its own over a period of time. The plasma membrane is also going to increase in its fluids. It's going to try to, the point is for the axon to try to regenerate and to prevent the cell body death. Now, if the connection of the severed axon is properly restored, because if protein is synthesized, and particularly in the peripheral nervous system, regeneration can occur. The axon can regeneration, these sprouts generate, the sprouts occur. So we're talking Schwann cells now. And the axon connects back to the distal part that's already broken. Then we're fine. Chromatolysis is going to end. The cell may return to normal functioning. So it is possible for the cell to return to normal function. But in the event that it doesn't, if the cellular swelling is not treated, cell death is going to occur. And then what's going to happen is your microglia is going to travel to the area where the cell is dying and try to engulf it. It's going to, so we call this necrotic tissue when the tissue begins to die. And then the neuroglia, microglia cells are going to travel, and even the astrocytes are going to travel so they can try to form scarred tissue. So phagocytosis is going to occur when there's going to be damaged tissue. That's something that you ought to know. Okay. All right. That's the axonal process. If the axon damage is close to the cell body, all the organelles are going to swell, and then the RNA is going to try to come in and produce more protein, and the cytoplasma is going to try to increase in its fluids in an effort to save the cell or the neuron. And to try to aid in the process of regeneration, particularly in the peripheral nervous system. In the central nervous system, the same thing happens. However, because in the central nervous system you don't have neurolemma, or you don't have the endoneurial membrane, then sprouting can occur, but the axons are never gonna, the proximal part of the axon is never going to connect to the distal part of the axon. It's just not going to occur. And that's what happens in stroke. That's what happens in problems with the CNS. Okay. All right. Let's do this. The Wallerian degeneration. Wallerian degeneration occurs when the distal part of the axon itself, the part that separates that's closest to the postsynaptic area, occurs. Survival of the axon is going to depend on three things: metabolic activity, cytoplasmic flow, and trophic substance or nutrition. But what happens is that part of the axon that is separated from the cell body, it's going to actually begin to degenerate within 12 to 20 hours. The axon itself is going to degenerate. It is going to degenerate before the myelin sheath. Now, when we have something like, Guillain-Barré syndrome, the Schwann cells that create the myelin, there's a problem. So the myelin degenerates in the peripheral nervous system. Okay. But in this case, the myelin can degenerate, but the axon might not degenerate. The myelin degenerates first in Guillain-Barré in the peripheral nervous system, and in the central nervous system, the myelin degenerates in multiple sclerosis in the central nervous system. The myelin degenerates, but the axon doesn't. The axon eventually will break down, but that's what causes the period of remission that we see in multiple sclerosis. The myelin is degenerating, but the axon is still functioning. It's just the transmission of the velocity of the impulse is slower. In Wallerian disease, the axon actually degenerates before the myelin sheath. Within 2 to 3 days, the connected muscles then become deinnervated or denervated, which means that they don't have nerve functioning, and so those muscles no longer work. Eventually, the axon is going to disintegrate, and its myelin is going to be broken into small pieces, and gradually everything will disintegrate. And that's it. And when that happens, microglia, phagocytosis is going to occur. It may take place anywhere between 3 to 7 days, or it might be completed in 3 to 6 months. Okay? And these are the added slides. So this is being recorded. I will put this into your class. I'll load this up into your class so you'll have these added slides. Okay? I wanted to talk about this process because it's important. What happens with myasthenia gravis, because we know what happens with, so if we have a problem with Wallerian degeneration, it means that the distal part of the axon degenerates before the myelin sheath. If we have a problem with, multiple sclerosis, myelin is going to degenerate in the central nervous system, as simple as that. If we have problems with Guillain-Barré syndrome, myelin is going to degenerate in the peripheral nervous system. If we have a problem with myasthenia gravis, then on the receptor site, you're going to have antibodies that break down the acetylcholine that binds, so it cannot bind good to the receptor site at the neuromuscular junction. And with that being said, what I'm going to do, those are the things that I need you to remember. And then, of course, you can have all types of tumors. And the only thing you need to know about the tumors are the ones that are most affected. Your brain tumors, these are uncontrolled growth, but the types of tumors you can have are astrocytomas. I won't ask you about them. Ependymomas and oligodendrogliomas. These are very malignant tumors of the brain, and these are tumors of those different types of cells. We've already talked about multiple sclerosis. So I want you to look very quickly for me, and then class will end. I'm going to the very end of this slide. What I need you to do is to look at these videos, one on multiple sclerosis, one on, and these are the names of the videos, okay? Multiple sclerosis, Guillain-Barré syndrome, and myasthenia gravis. Look at those videos on YouTube because class is over. It will help you to understand these disorders, and it's very simple. They're very short videos. So look at them. And what you need to review is the PowerPoints and look at your tables that I told you to look at. Okay? And do your readings. But basically, the way I function in quizzes is I take the words really from my PowerPoints or from the book. All right? Any questions, guys? Talk to me. Any questions? It's a lot. You need to study. But, and my quizzes are like exams. It's just that what I want to tell you about the quizzes is that they generally don't count but like 10%. So if 10% of 100 is only 10 points, so that allows you to do poorly on a quiz, and if you got like, let's say a 70, you got 7 out of 10 points. You're only missing 3 points. I always quiz first, multiple choice, so that you see how I'm going to quiz. But the midterm will count more and the final. The other thing you need to know about me is that I test comprehensively, which means that what you learned on the quiz in chapters 1 and 5, you cannot forget because you'll see it again on the midterm. So we will do the quiz, the first part of the class, and then we will do gross anatomy. Now, we are a little behind, but I don't mind being behind. The goal is for us to learn the materials, and it is hard material to learn. Okay. I don't know how many questions, but it could be what? 30, 40 questions. It's a quiz, but it's more like a test. All right. Don't worry about any assignments. The only thing you need to do right now is to study for your exams. Okay? Don't worry about assignments. All right? We'll talk about those later. If you've done your questions for Chapter 1, if you've been doing your coloring, excellent. Okay? You should already have drop boxes there, and you should be able to just put anything in the drop box you want to. I'd have to go out, but I'm pretty sure they're there. You will log into Blackboard for class like normal. You will take this quiz in eLearn. The quiz will open during class time, and I think it's there for about, I have to look at how long it's there because I can't remember, but I think it's a little over an hour. And then we will have class. Okay? All right, and look at these YouTube videos. In the meantime, I will be checking to see if you have questions. And I have to be honest, I'm so busy a lot of times that I may not necessarily attend to emails immediately. Okay. Let me see what's in the Dropbox. I have to go and look at it. But only thing I want to tell you, Lakin, is don't worry about that. Just be honest with you. The assignments I want you to do, you will get credit for it, but I don't really care. It's more important for me right now that you study and you learn this material. Okay? So don't worry about that. Nobody is penalized. Even though I give you deadlines for things to be done, I mean, I'm not penalizing you. However, if I say, now by so-and-so, if I come on this mic and say, have you put your stuff in there? then I'm telling you that what? That's when I absolutely need it. But right now, I could care less about the Dropbox or whatever. The assignments, if you're coloring, we haven't gotten to gross anatomy, and you would be coloring for gross anatomy. So don't worry about that. If you've done your questions for chapters 1 and 5 or whatever, remember I said do the questions, but do them for you. Okay? Any other questions? Okay, you don't know what you don't know until you go back and read and learn this, but you must spend time in this class. So you got this class, and what else do you have? What else are you taking? Are you taking RTIC? Okay. Yeah, you got to study for RTIC too. Oh yeah, but you hadn't gotten into your clients yet. You won't do that until July. If you're doing your observations, get them done. I think you can do 20 of them on by using the whatever, if those are what you need. But you have the option of coming on campus. You always have an option. Unfortunately, Tiffany, it would be good, but it's the way it is just the way the schedule runs, and we're a 12-month program. So any class you take is going to be pretty much loaded. But it is a lot. That's why we have 4-hour classes, and it is hard. I understand. Oh, the grant. Yes, guys, I am going to be funding. And I will, when I get back, we will start. I know that a lot of people have applied for grant funding, so we will go through the process. I've got a lot of people graduating off this summer. I will be funding in the fall. Okay? If you submitted an application to Ms. Matlock, then we will be funding. Okay. All right, any other questions? No. If you're out of state, guys, I'm sorry, I can't help. Not with my grant. You got to be a Tennessee resident for my grant. Other types of fundings you would have to get through financial aid or whatever. You guys have really been super. Hang in there. I promise you it's going to get easier. All right, thank you, Michelle, and good night, guys. Look at these videos. They will really help you. Okay, good night.